Welcome future doctors to this amazing platform of PW NEET English. Yes, I am Biswajit, your zoology mentor. So today in this video, we are going to complete the chapter from class 12, that is human reproduction, obviously the most interesting one. Now it's very important as well. It's not because it's interesting, it's because it's considered as a high weighted chapter. Now the chapter is laden with concept, so we have to learn every concept from the core. At the end of the video, you'll be getting DPPs and obviously practice sheets as well as handwritten notes of this particular class. Now from where you can get this, just check the link given in the description box. You have to click it and that will redirect to PW app. Yes, Physics Wala, the most loved and affordable platform for millions of students. Every year, every day, thousands are learning from this platform and going towards the path of success. So without delaying, we will be starting our today's lecture of human reproduction. So we are starting like this. There will be obviously an introduction of every chapter, and just after that we are going to talk about the anatomy of male and female reproductive system. Okay, these are comparatively easier one. Now we'll be coming to a conceptual topic, that is gametogenesis, that is spermatogenesis and oogenesis. Menstrual cycle, again a tricky, difficult but easy to learn topic. Fertilization and implantation, the sequential phenomena of embryonic development that we are calling pregnancy and embryonic development. The gestation period we are going to study, and it's going to be very interesting again. And we'll be concluding the chapter with parturition, the process of the childbirth or the baby delivery, and obviously the process of formation of milk and secretion, that is lactation. So as we know that we are Homo sapiens, human being, we are sexually reproducing animals, right? Have you ever seen that even a single human reproducing or following a sexual mode? No, right? And we are giving birth to young ones directly. That's why we are viviparous as well. Okay, if you observe birds, fish, then frogs, then what you can see that they are laying eggs. So egg laying animals, we are calling them oviparous, right? So now let's study the reproductive events in humans. Okay, so we are obviously unisexual or dioecious animal, means we are sexually dimorphic organism, as because in human being male sex and female sex is separated, and that's why we need to study the sexual mode of reproduction, because the reproduction is accomplished by the process of formation of gametes and their fusion. Okay, now when we are talking about gametes, there are two kinds of gametes: sperm or male gamete, ovum or female gamete. So the process will be initiated by the process of formation of gamete, that is gametogenesis. This process of formation of sperm and ovum. Okay, now what after gametogenesis? Specifically, this is the duty of male human being to transfer the sperms or male gametes into the genital tract or reproductive tract of female. So the process we will be calling it insemination. That is transfer of sperms into genital tract of female. Okay. Now what after insemination? Obviously, there is a particular location allotted inside the female reproductive system that we call the ampulla, or more precisely ampulla isthmus junction. There the sperm and ovum will get fused, and that fusion we are naming it fertilization. So what is fertilization? The fusion of sperm and ovum. Now what after fertilization? Just after fertilization, there will be formation of a single cell which is diploid in nature. You know, sperm and ovum, they both are haploid in nature. And for human, sperm is having 23 chromosome, ovum is having 23 chromosome. Now after fusion, they will produce a cell, that is zygote, with 46 chromosome. That's why we are calling it a diploid cell. Now after formation of the zygote, that will keep dividing. That process we are calling it the cleavage process in zygote, and that will be forming a embryo. Now when a particular stage called blastocyst will be formed in embryonic developmental stage, that blastocyst will get attachment, an attachment with inner uterine wall, and the process we are calling it implantation. So what is implantation? Attachment of blastocyst with uterine wall. Now what after implantation? Now we can say that the mother is pregnant now. So a new stage starts here, that is the pregnancy period or the gestation. So during the time of gestation, basically there will be sets of phenomena which we call embryonic development. Now in course of embryonic development, you can observe a formation of different organs, morphogenesis, and after that the process will end now. Now it's time for happiness in the family, because the fetus is going to see the light of the world. Okay, now the chintu or chint is coming. So that process of childbirth is called parturition. There is childbirth. Now the whole process I have summarized here in the form of some important diagrams. So here you can see that when the sperm and ovum is formed, the process we will be calling it gametogenesis. That is happening in separately in male reproductive system and female reproductive system. Okay. Now when they get fused, we are calling that they are fertilized, and the event is called fertilization. Okay. Now after fertilization, we can see there will be formation of a zygote which is diploid in nature, having 46 number of chromosomes. Okay. Now gradually the zygote will divide and will form the embryonic stage called morula, a bunch of mulberry fruits like structure, and eventually that morula will develop to form a football like hollow structure called blastocyst, and this blastocyst will get attachment with inner layer of uterus, that is with endometrium of uterus, and the process we are calling it implantation. And again after implantation, what we can see, the stages of development of the embryo inside mother's womb, right? And that stage we are naming it gestation or the pregnancy period, and the gestation will be terminated with the childbirth, that is called parturition. Okay, now coming forward to like if you compare the reproductive events, so these events were about reproductive events. Now if you compare reproductive events in male and female, you may find remarkable difference. You will find remarkable difference. So there are remarkable differences between the reproductive events in male and in female. For example, one example is given that sperm formation continues even in old men. See, there is spermatogenesis starts during the time of puberty in case of male, and it continues in old men even. But what about oogenesis? Oogenesis, the actual oogenesis process starts during the time of embryonic development of that female child, okay, even after the birth of her, right? And it continues till she is arriving at the age of 50 years. So at the age of 50 years, the process will be arrested there, the process will be ceases there, and that we call menopause. Okay, that we call menopause. Okay, now let's examine the male and female reproductive system in brief. Okay, so one by one we are going to study male reproductive system and female reproductive system. Now in a like normal manner, okay, commonly if you observe reproductive system, then you will be finding two kinds of organs are there: primary reproductive organs both in case of male and female, and secondary reproductive organs. Now, what do you call these primary reproductive organs? In male, one pair of testes, and in female, one pair of ovaries. We call them gonads. And what are their functions? Gametogenesis, gametogenesis is the function of gonads or primary reproductive organ. Testis, sperm formation will be there in ovaries, ovum formation will be there, and as well as there will be formation of sex hormones. Like for testis, it will be producing testicular hormones, and for ovary, it will be producing ovarian hormones. In male, androgens like testosterone; in female, progesterone and estrogens. So these two are the important function for primary reproductive organs or gonads. But what about the function of secondary reproductive organs? See, here in the secondary reproductive organs, you will be finding some genital ducts or associated ducts, associated glands, and external genitalia. And their function is transport of gametes, transportation of sperm and ovum, nourishment to gametes, providing nourishment or nourishing the gametes. So these two are the important function: transportation of gametes and nourishment, providing nourishment to the gametes. Okay, you have to remember that the secondary reproductive organs, they cannot produce gametes, or they cannot even produce hormones. They do not have the endocrine property. Okay, so here the learning outcome till now will be solving in the form of two questions. Okay, so question 1, here you can see that human being is, are we sexually dimorphic? Yes, we are sexually dimorphic animal. Are we sexually monomorphic? No, we are not sexually monomorphic. Hermaphrodite, bisexual animals are sexually monomorphic. Are we dioecious? Yes, we are dioecious. The opposite one is monoecious. Hermaphrodite, again, the bisexual animals are monoecious. We are dioecious. So the answer is both 1 and 3. Now let's see second question, saying that implantation is attachment of what into the uterine wall? Yes, implantation is the attachment of a particular embryonic stage that we call blastocyst into the uterine endometrium. Okay, now let's start with male reproductive system. Okay, so the male reproductive system is located, where it's located? Male reproductive system is located into the pelvic region. What is pelvic region? You can say it's a lower abdominal region. Okay, you know the region very well. Now let's make a table or summary that what are the anatomic structures associated with male reproductive system. Okay, male reproductive system. We have primary reproductive organs, we have secondary reproductive organs. Now primary reproductive organs, we have one pair or two tests, in singular TES, TIS. Okay, and what will be the function of this test is spermatogenesis, that is sperm formation. So factory, the test is there, the factory of sperm. Okay, and obviously formation of male sex hormone that we call androgens. The prime androgen is the testosterone, and they are kind of steroid hormone. See, remember this word. Either you are calling, you are talking about the male sex hormone, or you are talking about the female sex hormone. All of them belong to the group, all of them belonging to the group of hormone that is called steroid hormone. Okay, so for male, their androgens, a group of hormone, and the prime androgen is testosterone. Okay, what kind of hormone are they? Steroid hormone. Okay, now we are coming to the secondary reproductive organs in males. We have some accessory ducts, we have some accessory glands, and we have external genitalia. External genitalia means the organs which are visible mainly from outside. Okay. So, what are the parts associated with the accessory duct for male reproductive system? They are retitestis, vasa efferentia, epididymis, vas deferens, ejaculatory duct, and urethra. So these ducts are under accessory duct system. Now, what are the accessory glands? We have seminal vesicles, prostate, and bulbourethral gland. Remember, we have two seminal vesicles, we have one prostate, and we have two bulbourethral gland. And what about external genitalia? When you talk about external genitalia in human being, in human male, obviously the copulatory organ penis and scrotum, a skinny sac which is present outside the abdomen, whereas this testis, you will be finding the testis inside the skinny sac which you call scrotum. Okay, now I think we have summarized in this tabulated format. Now we'll be detailing it. Now here we can see that two diagrams of male reproductive system is given from two different angle of view. This is frontal view, this is sagittal view. This is frontal view, this is sagittal view. Now we have to label them. Okay. Now we are starting from testis. After testis, outside the testis, you can find a coiled portion, that is epididymis, and the testis is safe inside the skinny sac that we call scrotum. Okay. Now let's track the way of epididymis. Now from the tail region of epididymis, you will find that from the tail region of epididymis, a tube is going upwards towards the abdominal cavity. So this tube we call it vas deferens. Now this vas deferens will go towards the abdominal cavity and it will take a U-turn, or it will loop over near to the ureter, the junction of ureter and urinary bladder, and again we will start, again the vas deferens will start coming down, it will start descending down from abdominal cavity, taking a looping over or U-turn near to the ureter and urinary bladder junction. So this is urinary bladder. And it will join with the duct which are coming from seminal vesicle. So we can see here into different angle the vas deferens joined with the seminal duct coming from the seminal vesicle and forming ejaculatory duct. So how ejaculatory duct is formed? The ducts which are coming from seminal vesicles. So duct of seminal vesicle, we are calling them seminal duct. Seminal duct plus vas deferens forming ejaculatory duct. Remember it. Okay. Now in this particular location where this ejaculatory duct is formed now what will happen? Duct will also be coming from the urinary bladder, that we are calling urinary urethra, and here you will find a single gland, that is prostate gland. Now from prostate a duct will come, that is prostatic urethra. Now the duct from urinary bladder, the duct from urinary bladder, that is urinary urethra, the duct from prostate and the ejaculatory duct will conjoin together to form a common duct that we are naming urethra. Urethra. Also, we call it urinogenital duct, as because through urethra, urine will be released out. Through urethra, semen or the sperm will also be released out. So that's why it's a common passage for sperm or semen and urine. That's why we are naming the urethra in case of male urinogenital duct. Okay. Now the urethra will proceed towards the copulatory organ of male, that is penis. The copulatory organ of male, that is penis. Here you can see this is penis. And the urethra is passing through penis. Now before entering into the penis, the urethra will receive secretion of two more glands, the alkaline mucus secretion of two more glands. We call them bulbourethral gland. We call them bulbourethral gland or Cowper's gland. Also it's known as Cowper's gland. So here I am marking the bulbourethral gland, just attaching with the urethra, just before the entry inside the penis. Here also you can find these bulbourethral glands or Cowper's glands. They are secreting alkaline mucus, helping the penis for lubrication. Okay. Now the enlarged end of penis you will be finding here, and within this enlarged end of penis will get opening of urethra, that we are calling urethral meatus. Or urethral opening, urethral meatus or urethral opening, and the enlarged end we are naming it glans of penis or glans penis. And you can see here the penis is covered by a retractile skin. So the retractile skinny covering of penis, you can call it piprius or foreskin. The retractile skinny covering of penis, you may call it foreskin or piprius. So in short, that was all about the anatomical structure of male reproductive system. Okay, now we'll be moving forward. I'm decorating this slide a little bit. Okay, now let's get detailed knowledge about our primary reproductive organs, that is testes. Okay, so when someone will ask you, sir, what kind of organ are they? So you need to tell them that testes, they are extra abdominal organs. Means they are not present inside our body, they are present outside the abdominal cavity, and they are situated, where they are situated? Inside pouch called scrotum. Okay. So if this is the body cavity of human being, so testes, you will be finding them outside the scrotal pouch, right? Or scrotal sacs. though it has a connection with abdominal cavity, but this present outside the abdominal cavity. So this is the body cavity of us. The upper one cavity is thoracic cavity and the lower cavity is abdominal cavity, and the lower abdominal cavity is also can be called as pelvic cavity. Okay. So this is scrotum, and scrotum is divided into two scrotal sac. Inside each scrotal sac you will be finding one testis, and the connection of testis with the abdomen, we are calling it spermatic cord. The connection of testis with abdominal cavity, what we call it, spermatic cord, right? And basically what happens during the time of embryonic development, you won't be finding testis inside the scrotum. During the time of embryonic development, you will be finding testis inside your abdominal cavity. Now in the course of development, near about when the male fetus is 7 months old, the testis will start descending down into the scrotum, and a particular fibrous tissue will be guiding them to this descending process, and this fibrous tissue we are calling them gubernaculum. Okay, though it's not given in NCERT, so I am not detailing them. Okay. Now you have to remember this one, spermatic cord, and obviously sometime like particular disease condition is asked. Sometime you will be finding, in very rare case, some male, they don't have testis present inside their scrotum. In their case, the testis that present into their abdominal cavity, and the testis have failed to descend down, and that disease we are calling it cryptorchidism. And a tunnel is connecting the abdominal cavity to the scrotum, and the tunnel we are calling it inguinal canal. Right? Okay. Now that's a different thing. Now the question is asked, the important thing, that why the location of testis is extra abdominal inside the scrotum. So the answer is, scrotum maintains the temperature, low temperature for spermatogenesis. So what is your body temperature? Your body temperature is 37 degrees Celsius, right? Now for the process of sperm formation, you need -2 to 2.5 degrees centigrade less than our body temperature, that is near about 34 to 35 degrees Celsius. That cannot be maintained inside the abdominal cavity. So that's why Almighty has like given us this advancement to maintain low temperature, which is necessary for sperm formation. Okay. The next thing, what is written here? Now we'll talk about that the testis, they are really delicate organs, right? Now you need to protect them from any kind of friction or shock while you are running or moving or doing exercise. So we'll talk about the covering of testis. So the main covering of testis, you can see here, it's tunica albuginea. This is the main covering of testis. This is the middle layer of the testis, which is also acting as shock absorber. And along with tunica albuginea, there are other important layers are also there. The outermost layer of testis, the covering layer of testis, that is called tunica vaginalis. Then you will be finding tunica albuginea, and the innermost layer of testis, that we call tunica vasculosa. So altogether there are three covering or layers of testis you will be finding. So the important one is tunica albuginea and the other two, tunica vaginalis, the outer one, tunica vaginalis, this is the outer one, and tunica vasculosa, that is the inner one. And the middle one is obviously tunica albuginea. Now what about the dimension of testis? That's also given here. The length of testis is 4.5 centimeter. Width of testis is 2 to 3 centimeter. And if we talk about the weight of testis, in an average human being is near about 25 grams. So that was the basic dimension of testis. Okay. Now here I would like to elaborate a bit. Suppose this is the scrotum that is divided into two chambers. One scrotum is having two scrotal sacs. Now here you will find your testis. The comparative location of your left and right testis is little bit uneven, and the connection of testis with abdominal cavity, which we are naming it, what we have named it? We have named it spermatic cord. That includes nerve, blood vessel and vas deferens, also lacteals. Okay. Below the testis, the fibrous tissue, you call it gubernaculum, and you will find that there is seasonal change in the position of testis. In winter, we will feel that testis is the scrotal sac, they are closer to your body. In summer, we will find that the scrotal sacs are loosen up. So why it's so? So there will be position adjustment according to your body temperature and environment, and that is done by two important muscles associated with your scrotum. One is dartos muscle and another is cremaster muscle. Okay, now we'll get inside each testis. Now when we talk about one testis, they are compartmentalized into 250 chambers that we call testicular lobules. So if question is asked that each test is having how many chambers or how many testicular lobules, we will answer 250. If question is asked that human body has how many testicular lobules, then you will answer 250 into 2, that is 500 testicular lobules. That means in human body there are two testes, right? Okay. And inside each chamber or inside each testicular lobule, you will find 1 to 3, it can be 1, it can be 2, it can be 3 highly coiled tubes, the actual factory of spermatogenesis, that is seminiferous tubules. Okay. So let me elaborate it. Suppose this is scrotal sac. This is the covering of testis, tunica albuginea, and that will be making the septum, or the compartmentalization of testis is done by the layer tunica albuginea. Here you will find gubernaculum, and outside the tunica albuginea you will get a bilayered tunica vaginalis, which is again an incomplete layer, and inside each chamber the inner lining of chambers are lined by tunica vasculosa. Now you don't have to remember all these things, just have to understand what is the basic covering of testis, that is tunica albuginea, and they are forming the testicular the septum to form the testicular lobules, and like this 250 chambers are there. Okay, and inside each chamber what you will find? You will find highly coiled tubular structure called seminiferous tubule. Now the ducts which are coming from seminiferous tubule will come outside and will form a network inside the testis. We'll call this network as retitestis, and from this retitestis you can find origin of 15 to 20 or 30 ducts outwards to the testis, which we are naming vasa efferentia, and all the vasa efferentia will combine together to form another highly coiled tubular structure, epididymis, and the tail of epididymis will give rise to vas deferens. Okay, so each chamber we are naming it testicular lobule. How many lobules are there? Total 250 testicular lobules in each testis. And these highly coiled tubes, what we are naming them? These highly coiled tubes, we are calling them seminiferous tubule. In each testicular lobule, you will be find 1 to 3 seminiferous tubule. So in a testis, average 500 to 750 seminiferous tubules are present. And for your information, the seminiferous tubule, they are the location for spermatogenesis, sperm formation. Okay. Now from seminiferous tubule, after seminiferous tubule, what you are getting? You are getting these straight tubes, we are calling tubuli recti, and all these tubuli recti will combine together to form this network, and that network is called vasa efferentia. Sorry, this is retitestis. I'm extremely sorry. Retitestis from retitestis these ducts are coming out, we call them vasa efferentia. And the vasa efferentia will join together to form this highly coiled tube that we are calling it epididymis. And from the tail region of epididymis or from the cauda epididymis, there is origin of vas deferens. So, if there is tail of epididymis, there will be head of epididymis, there will be body of epididymis. So, epididymis is divided into three portions: head, body and tail. Head is called caput epididymis, body is called cauda epididymis, corpus epididymis, tail is called cauda epididymis. Caput epididymis, corpus epididymis, cauda epididymis. And from cauda or tail epididymis, there is origin of what? Origin of vas deferens or sperm duct. Now I think this slide is very much clear. Now what about inside the seminiferous tubule? Inside the seminiferous tubule, you will be finding two types of cell. There is one type of cell, we are calling it male germ cell. Also we call them spermatogonia. Those who are responsible for sperm formation. Basically, this cell will divide through the process of meiosis and will produce sperm or spermatozoa. And the second types of cell, which are called Sertoli cell, also we are naming them nurse cell or sustainocyte cells, which are columnar in nature, tall slender cells, which are called Sertoli cell. They are responsible for providing nutrition to the germ cell. So let's look inside seminiferous tubule. Okay. So when you are talking about seminiferous tubule, everything inside them we are going to study in detail. Now what we are naming the space outside the seminiferous tubule. So the space outside the seminiferous tubule, we are calling it interstitial space. Interstitial space, the space outside the seminiferous tubule, we are calling it interstitial space. Okay. Both inside and outside the seminiferous tubule, they are important for us to study. Okay. So that's why it's written here, region outside seminiferous tubule, we call it interstitial space. And what are the structures we are gonna find here inside the interstitial space? Remember, small blood vessels, not large blood vessels. Some interstitial space has small blood vessels, Leydig cell, they are the cell responsible for the sperm production, also we call it interstitial cell. Leydig cells are also known as interstitial cells. They are responsible for production of androgen hormones, which are responsible for sperm production. And inside that also you will find immunologically competent cells. Basically they are nothing but WBC, immunologically competent cell, ICC. Okay. Leydig cell or interstitial cells synthesis and secretes testicular hormone, that is androgen, and the primary androgen is testosterone. And obviously you cannot forget it that they are steroid group of hormones. Now this diagram, obviously you will find into your NCERT book. We have taken a chunk of testis and we are observing or doing histological study of that. So these parts are basically a single seminiferous tubule, seminiferous tubule, seminiferous tubule, seminiferous tubule, seminiferous tubule, seminiferous tubule, seminiferous tubule. And the part outside that, this is interstitial space, right? So this is a single seminiferous tubule. I'm writing in shortcut ST, seminiferous tubule, and the space outside, that is interstitial space. Okay. Now what I have done here, I have taken out one single seminiferous tubule and the associated interstitial space here. So if you observe a single seminiferous tubule, what you are going to find? So the outermost layer of the seminiferous tubule, that we are calling it basement membrane or basal lamina. Just inside, if you peep inside the basal lamina, the cells you are getting here, these are forming the germinal epithelium, or these are the germ cells. And these germ cells, we call them spermatogonia. And these tall slender cells, we call them Sertoli cell. Okay. Now inside the lumen of this seminiferous tubule, what you will find here? You will find the developing stages of sperm, that is spermatogenesis is observed here. Towards the lumen you will be finding mature sperm attached with the Sertoli cells. Okay. Now what about this interstitial space? This space is called interstitial space. In interstitial space you will be finding this particular important group of cells, and we call them Leydig cells or interstitial cells This Leydig cell and interstitial cell, they are responsible for production of androgen, or the prime androgen is testosterone. Okay. Now eventually the testosterone will diffuse inside the blood, and some of the testosterone will enter inside the seminiferous tubule and will take part in the development of male reproductive system, and obviously the sperm production, and obviously the appearance of beard, mustache, what we call secondary sexual characteristics. For all those things, the testosterone is very much important. Okay, Now what are these? These are the small blood vessels. And this particular cells we are calling immunologically competent cells. They are basically WBCs. So in interstitial cells, you cannot like forget their name. Immunologically competent cells, small blood vessels along with Leydig cell. Okay. Now the role of the Sertoli cells are very much important here. Why? See, the sperm production is going inside, and this sperm or spermatozoa formed inside the seminiferous tubule, they are haploid in nature, having only 23 chromosomes. And for our immune system, haploid cells are foreign cells. So if there is direct contact of blood with your haploid cell inside the seminiferous tubule, the immunological components like WBC and macrophages, they will kill the haploid cell or the spermatozoa inside your seminiferous tubule. So for that, you need to make a blood testis barrier. Now what is blood testis barrier? The separation boundary which is basically tight junction. Which will stop the leakage of blood, which will stop the leakage of blood inside the seminiferous tubule. And where you will be finding this tight junction? Yes, these tight junctions you will be finding within the Sertoli cells. So there is another important function of Sertoli cell to form the blood testis barrier. That is separating the internal environment of seminiferous tubule from blood or from immunological cells of blood, so that the haploid cells will get survivability. That's all. Now we'll talk about the hormonal regulation upon our reproductive system. Hormonal regulation is a very important topic. Again, each and every concept of this chapter is important. So we know that pituitary is the master, and who is master of master? You know very well, hypothalamus. Yes. So if you are talking about hormonal regulation, the regulatory message or signal will be originated from hypothalamus. Now here hypothalamus is releasing gonadotropin releasing hormone, GnRH, gonadotropin releasing hormone. The GnRH will give positive signal to anterior pituitary or adenohypophysis, the actual glandular part of your pituitary. So the hypothalamus will release a message in the form of hormone, gonadotropin releasing hormone, GnRH, that will stimulate the cells from inside the anterior pituitary, and upon stimulation, anterior pituitary will release two hormones here. Combinedly we call them gonadotropins. One is FSH, one is LH. FSH for follicle stimulating hormone, LH for luteinizing hormone. Now in case of male, the LH is also called as ICSH, interstitial cell stimulating hormone. Yes, because the LH, their target cells are nothing but Leydig cell. The target cells are what? Leydig cell. LH is targeting Leydig cell. Okay, and the other name of Leydig cell, you know, interstitial cell. So also in case of male, LH or luteinizing hormone is known as ICSH, interstitial cell stimulating hormone. Interstitial cell stimulating hormone. Okay. Now on stimulation, Leydig cell will release the male sex hormone on the testicular hormone, that is testosterone, in the form of androgens, and obviously I am reminding you again and again, they are steroid hormones. Androgens are steroid hormones, so as testosterone. Okay. Now, what is the function of this testosterone? Again, I am recapping it once again. Testosterone helps in spermatogenesis. Testosterone helps in the development of male reproductive system, the accessory ducts and accessory glands. Testosterone helps in appearance of beard and mustache, muscular body in case of male, that is help in development of secondary sexual characteristics feature. Okay. Now, what about the other gonadotropin, that is FSH, follicle stimulating hormone? The FSH will stimulate Sertoli cell. Remember, what is the other name of Sertoli cell? Nurse cell or sustenticular or sustenocyte cell. Now, these Sertoli cells are responsible for the survivability of the germ cell and obviously for the process of spermatogenesis. How? See, the Sertoli cell, they are releasing some hormones called ABP, androgen binding factor, releasing hormones like AMH, anti-Mullerian hormone, releasing hormones like SMF, sperm maturation factor, releasing hormones like inhibin. Okay. Now, one by one we are going to learn their function. Inhibin, that is inhibitor of FSH. So, if there is more amount of FSH present into male blood, or if titer of FSH is higher, the Sertoli cells are overactive, then the Sertoli cells can release inhibin to give negative feedback to the anterior pituitary and hypothalamus that will cease production of FSH. So, inhibin, they are inhibitor hormone for follicle stimulating hormone. Okay. Now, what is the function of ABP, androgen binding protein? Again, important concept is here. A seminiferous tubule, Sertoli cell. And what are these cells? Spermatogonial cell or germ cell, right? Okay. Now, who is producing testosterone? Leydig cells producing testosterone. Who will be stimulating Leydig cells? Luteinizing hormone or ICSH. Interstitial cell stimulating hormone will be stimulating Leydig cell. Leydig cell will be producing testosterone. Testosterone have some other works like development of male reproductive system, development of secondary sexual characteristics features like beard and mustache. Testosterone also helps in the process of hematopoiesis or erythropoiesis. That is the reason male are producing more amount of testosterone. That's why we have higher volume of RBC and blood and hemoglobin produced inside our body in compared to female. And obviously testosterone is needed for the process of spermatogenesis inside the seminiferous tubule. Now, how the testosterone will come from interstitial space towards the seminiferous tubule? You need to give some invitation. Now, here these Sertoli cells, they act by releasing androgen binding protein, and these androgen binding proteins are expressed here, and the testosterone will come and bind with the cells of seminiferous tubule, and that will promote the division of this germ cells or spermatogonia towards the sperm formation. Okay. Now, sir, what is the function of AMH, anti-Mullerian hormone? See, basically the function of this hormone is required during the time of embryogenesis, that help in determination of sex of male. Okay, just know this much. Anti-Mullerian hormone that help promote formation of male reproductive system. Anti-Mullerian hormone. Sperm maturation factor, SMF, what is their function? See, on course of spermatogenesis we will get to know that the last step, that is formation of spermatozoa or sperm cell from spermatid cell. So for the time being, you just know this information. Sperm maturation factor, SMF, sperm maturation factor, they are responsible to complete the last step of sperm production. Without them, the sperm will remain as immature spermatid. The sperm will not get maturity if you do not have sperm maturation factor. Okay. Now let's move forward. Now we'll talk about male accessory glands. Okay. Now we know there are three types of glands associated with male reproductive system, and what are they? They are one pair of seminal vesicle, single prostate, and one pair of bulbourethral gland. Also, they are known as Cowper's gland. Okay. Now, altogether, they will be secreting a liquid which we call seminal plasma. Seminal plasma, the secretion of the accessory glands of male reproductive system. Okay, and this seminal plasma that is rich, it is having some important factors which are needed for the nourishment and survivability of sperm, that is fructose. Fructose, the main source of energy for sperm. Okay, because sperms, they always swim, they need large amount of energy and they harvest energy by breaking down the fructose. Now if you ask me, sir, if you give glucose in case of fructose, will it work for sperm? Obviously it will work, but for our body system, sperm will get only fructose. Okay, and fructose is produced by this seminal vesicle glands only. Okay, also they are rich in calcium, that is helping maintaining alkaline pH. Acidity is toxic for sperm, and it also has some important enzymes for survivability of sperm inside the female reproductive system just after insemination or coitus. Okay. Now, especially the function of bulbourethral glands, or the secretion which is alkaline secretion, the secretion of bulbourethral gland, it is basically alkaline mucus secretion that helps the penis for lubrication, easy passage of semen, Okay, who helps that? the secretion of bulbourethral glands or Cowper's gland, and also that helps in neutralizing acidity of urine in the urethral passage. So these two are the very important function of bulbourethral secretion, as because that is alkaline. So when like semen is about to come out or sperms are about to come out through the urethra, the passage is already impaired by the stresses of urine, right? And the urine is highly acidic, is having acidity of 6. So until unless you are neutralizing the passage, okay, you are giving a alkaline passage toward the sperm may die inside your urethra. So that's why that role is played by the secretion of the mucus secreted by bulbourethral gland. Now we have two external genitalia. One is penis, another is scrotum. Why external genitalia? Is because they are visible from the outside. Okay. Now, how is the what is the structure of penis? They are made by special erectile tissue. Penis, they need erection just prior to copulation or insemination, and this special erectile tissue, we name it corpora cavernosa and corpus spongiosum. They are two important erectile tissue associated or present inside your penis. The enlarged end of penis is called glans penis, and the glans penis basically made by corpus spongiosum, remember it, and the urethral passage is also will be through corpus spongiosum. So that's why it's a very important part. And the penis is covered by a loose folded retractile skin, and we call it pepreous or foreskin. Okay, now let's talk about the penis. Okay, so this part is called shaft of penis, and this part is called glans of penis. Okay, towards the dorsal surface of penis, this is the dorsal side, this is the ventral side of penis, you will find two erectile tissue, that is corpora cavernosa. How many? Two corpora cavernosa. And the urethral passage, that is surrounded by the tissue of corpus spongiosum. Okay, remember it. So the urethral passage is through corpus spongiosum, the urethral opening through corpus spongiosum, even the glans of penis is also through corpus spongiosum. Now let's talk about scrotum. Scrotum is connected to abdomen by spermatic cord. Spermatic cord that is made by blood vessel. Nerve, vas deferens and lacteals. And during the time of embryogenesis, the canal through which the testis descends inside the scrotum, that canal is called inguinal canal. And the movement, seasonal movement of scrotum depending upon the temperature is controlled by these two muscle, dartos muscle and cremaster muscle. So here some questions for you. So you can find here seminiferous tubules contain dash cells for providing nutrition to sperm cell. Seminiferous tubule have Sertoli cell for providing nutrition to sperm cells. Okay. Another question, assertion and reason. Let's solve it. Assertion is saying that tests are located outside the abdominal cavity within the scrotum. Absolutely true. The reason is saying the scrotal muscles help to regulate a lower temperature in the testis. Yes, the dartos muscle and the cremaster muscle, we are saying the position adjustment of the scrotum or scrotal sacs depending upon the body temperature and the external temperature, which is essential to maintain the low temperature, that is essential for spermatogenesis. Yes, that is also very true. So the extra abdominal location of testis inside the scrotum is due to make it necessary for spermatogenesis. So answer will be both assertion and reason are true, and reason is correct explanation of assertion. Now let's move forward to the female reproductive system, right? Female reproductive system. Okay. Now when you talk about female reproductive system, like male, they also have primary reproductive organ and secondary reproductive organ. So let's see what's written here. For female reproductive system consists a pair of ovaries, right and left ovary, along with a pair of oviducts, uterus, cervix and vagina. So they are the part of accessory ducts, oviducts or fallopian tube, uterus, cervix and vagina. Ovaries, they are the primary reproductive organ or gonad, and external genitalia located into the pelvic region. Now the external genitalia part for female reproductive system, we call it vulva, right? Okay. Now obviously we know that ovaries, they are responsible, for the formation of ovum, or that is called oogenesis. Ovaries also responsible for formation of ovarian hormones. They are estrogens. See, the spelling of estrogen can be OES or simply ESTROGENS, and progesterone. These are the main hormones produced by ovaries. There are some other hormones like ovaries can produce relaxin, ovaries can produce inhibin as well. Okay. Now these parts of the system, along with, now we have to incorporate another two glands. Okay, while we are studying female reproductive system, a pair of mammary gland, this is the exclusive feature of mammal. See, if someone asks you, do we have mammary gland? No. As a male, yes, I also have mammary gland, but that is non-functional or vestigial. Okay, if you are answering no, so that will be a wrong answer. For mammal, both the sexes, male and female, they have mammary gland. The difference is there. The difference is the females, they have functional mammary gland. Okay, males, they have non-functional or vestigial mammary gland, but the presence of mammary glands are there both in case of males and females. Okay. Now, along with the pair of mammary glands, as because they're integrated structurally and functionally to support the process of ovulation, fertilization and pregnancy, birth and child care. Now, why we need to talk about mammary glands? Whenever we are studying the whole and soul phenomena associated to female reproductive system, we need to talk about child care, that is formation of milk, lactation and secretion, okay, that is after the childbirth. And obviously these phenomena are somehow related to the endocrine system and the emotion of mother. That's why mammary glands are also incorporated inside the female reproductive system. Okay, now let's talk about the anatomy of female reproductive system. Again, this view is a sagittal sectional view, like from side. This is the sagittal section. From side way, like we'll be studying what are the parts of female reproductive system. See, the female reproductive system, the parts whatever we are going to study, they are located behind this particular structure. That is pubic symphysis. What is pubic symphysis? Pubic symphysis is a cartilaginous joint of the coxal bones or the pelvic bones. Okay, so behind pubic symphysis, here you are getting your, like in case of female, this is the urinary bladder and the urethral passage and the urethral opening. This is the urinary bladder. And here you are getting the passage of urine through urethra. And here you are getting the urethral meatus or urethral opening, okay, near to the external genitalia of female. This zone we are calling it vestibular zone. Okay, so this is the dorsal side, sorry, this is the ventral side of the body and this is the dorsal side of the body. So towards dorsal side you are getting urethral meatus or urethral opening. Now ventral towards the urinary bladder you are getting the female reproductive parts, important female reproductive parts. Okay, so what about this structure? The womb or uterus, where the development of the baby and the menstruation every month taking place. Uterus is the uterine cavity. Okay, now this neck region of the uterus, we call it cervix. The region we are calling it cervix, and the zone here we are calling it cervical canal, and after cervix we are getting here vagina and all together this cervical canal and vagina, we call it birth canal. Because during the time of parturition, the baby will come out through this passage, through this canal, right? Now here you are going to get the opening of your vagina, that is the vaginal orifice or the vaginal opening, and where you are going to get the vaginal opening, just ventral to the urethral opening, and the zone here we are calling it as vestibule. Vestibule. Now if you check the vestibule properly, you are going to get some external genitalia here. So the outermost one, so the outermost structure, we are calling it mons pubis, which is a fatty cushion like structure. Inside the mons pubis you will get a fleshy fold that is called labia majora. Inside the labia majora we will get labia minora, and towards the upper junction of labia minora we will get a finger like structure which is just homologous to penis. The structure we are naming it clitoris. Okay. And sometime you also may find here that the opening of vagina that is guarded partially by a thin membranous structure which we call hymen. The opening of vagina or vaginal orifice. Okay. Now let's get a detailed idea about the ovary. Okay. So when we are talking about the female gonad or ovary, the function is female gamete formation and production of the steroid hormones like estrogens and progesterones. For that, dedicated cells are there inside the ovary. Okay. The dimension of ovary, 2 to 4 centimeter in length. What is the location of the ovary? So if this is the uterus, both the side of uterus will be getting 2 ovaries are present and attached by ligaments like structure. Okay. So the ovaries are connected to pelvic wall and uterus. So it has two-way connection. It has connection towards the uterine wall. It has connection to the pelvic wall or pelvic cavity. With the help of what? With the help of ovarian ligaments. Ovarian ligaments, the fibrous tissue. Okay. So this is another view of female reproductive system. Obviously, we'll study in a detailed manner. Now, first complete the discussion of ovary. Now we are taking example of one single ovary. So if this is a single ovary, this part is called ovarian hilus or ovarian hilum. This will be the entry point of blood vessel and nerves. Okay. And the outermost layer of ovary, that is called ovarian surface epithelium. You'll be finding some cuboidal cells here. So this is the outermost layer of ovary. What do we call? That is ovarian surface epithelium. Okay. Now below the ovarian surface epithelium, you will find a tissue or covering of dense fibrous irregular connective tissue that we call tunica albuginea. Inside the tunica albuginea, the place we are getting that we are calling it ovarian stroma. Okay. Now again this ovarian stroma, they are divided into two sections. The inner zone of ovarian stroma, where you will be getting the blood vessels and nerves, that we are calling ovarian medulla, and the outer zone of ovarian stroma, where you are finding certain developing stages of follicles. This space we are calling it cortex. So ovarian stroma has two zones: ovarian cortex, that is the outer zone, ovarian medulla, that is the inner zone, where you will be finding nerves and entry point of nerves and blood vessels. So here inside this medullary zone, where you will be finding which part of ovarian stroma having the developing follicular stages, you will be finding inside the cortex zone. Okay. Okay, so this is a frontal view of female reproductive system in detail. So let's study this. So one by one we are going to study it. So this part, this inverted pear shaped structure, we are calling it uterus, right? We are calling it uterus, and this upper part of uterus, we call it uterine fundus. Okay. Inside the uterus, this is called uterine cavity. The whole uterus also we call it womb. Okay. Outside the uterus, this non-glandular tubular part, we are calling it vagina. Remember, the vagina is a non-glandular part, no glands are present there. This is the vaginal opening or vaginal orifice, and the narrow channel which is connecting the uterine cavity to vagina, where you will be finding some mucus plaques. This is called cervical canal, and the zone is called cervix. Right? Okay. Now, both the side of the uterus, here you are getting your left ovary, here you are getting your right ovary. This is left ovary, this is your right ovary. Okay. Now, very interesting fact I am telling here. So, at a single point of time, both the ovaries are not reactive. Suppose in this month, your left ovary is active, that will be releasing one single secondary oocyte during the time of ovulation. So the very second month, your right ovary will become active and that will ovulate or that will release another secondary ovary. So ovaries, their turn is like that. Alternative months they will become active and they will ovulate one single secondary oocyte. Okay. Now this arm kind of structure, what we are calling it? This arm kind of structure, we are naming it fallopian tube. Also you can call it oviduct. Also you can call it uterine duct. Now when you are observing this fallopian tube, this funnel kind of structure, we call it infundibulum. This wider part we call it ampulla or ampulla. This narrower part we call it isthmus. So you can say that fallopian tube has three parts: infundibulum, ampulla and isthmus. Which is the wider one? Ampulla is the wider part. Okay. And here you can find the finger-like structure associated with the infundibulum. So what is the function of this finger-like structure? When there will be ovulation, the ovary will kick out one single secondary oocyte out of it, like developing stage of the ovum. Then, like the wicket keeper in the cricket match, with the help of the finger and gloves, they will try to catch the ball. So here this infundibulum having finger-like structure, we are calling them fimbri. With the help of this fimbri, they will catch the secondary oocyte and channelize inside the lumen of fallopian tube. Okay, and inside the fallopian tube, you'll find a special kind of tissue or cells with cilia, that is ciliated epithelial cells, ciliated epithelium, ciliated columnar epithelium. Why it's so? Why the fallopian tube is equipped with a ciliated cell? Is because the ovum, the maturing ovum, cannot swim or cannot travel by its own. It's immobile in nature. So this dancing cilia will be like a football match. Football players, they're kicking the football towards the goal, right? So what is the goal for this ovum? The goal is to meet with the sperm to the ampullary region or ampullary isthmus junction. So this ciliary movement will channelize the immobile ovum towards the ampulla or ampulla isthmus junction. Okay. Now let's talk about the wall of uterus. So if this is a female uterus, so we are going to study the uterine wall. The outermost layer is called perimetrium. The middle layer is called myometrium. And the innermost layer is called endometrium. Right? So there are three layers of the wall of uterus. Metrium means wall of uterus or uterine wall. Peri means this is the outermost layer. Endometrium means this is the innermost layer. Myometrium, that is a muscular layer, which is the middle layer. Okay. Now let's study the theoretical part of uterus, whatever we were observing with our diagrams. Okay. So shape of uterus, there is inverted pear fruit shape. Also we call it womb, where the fetal development will be taking place. Okay. The uterus is attached to the pelvic wall with the help of some uterine ligaments. It has three important layers. What are they? Perimetrium. Perimetrium is a membranous layer, thin membranous layer. Made up of connective tissue and squamous epithelial tissue. The myometrium is a layer made up of smooth muscle which responds like when there will be secretion of oxytocin hormone, it helps for uterine contraction. The myometrium, the strong contraction of myometrium during the childbirth smoothen the process. Okay. And obviously the last layer, innermost layer, endometrium, which is a glandular layer. And when you talk about this endometrium, the cyclic change, that is menstruation, takes place from the functional layer of the endometrium, and also endometrium is the site of implantation of blastocyst or the site of pregnancy. Okay, now let's talk about the female external genitalia. What are the structures associated? Mons pubis, labia majora, labia minora, and hymen clitoris. So let's summarize them. So the first structure is mons pubis. Remember, what is mons pubis? Fatty cushion with skin and hairs, pubic hairs, the outermost part of female external genitalia. Inside the mons pubis, there is a fleshy fold that is called labia majora or major lip. Remember this word, fleshy fold, which is present just beneath the mons pubis. Now below labia majora you will be getting the minor lip or labia minora, thin fold of skin, thin folded skin under labia majora. Okay. Now the next structure which will be arising from labia minora, that is clitoris. Finger-like structure. the upper junction of labia minora. And the last structure is hymen. Thin membrane which covers the vaginal opening partially. So the vaginal opening is partially covered by hymen. It cannot be completely covered. So if it's completely covered, you can imagine that during the time of menstruation, how the menstrual flow or menstrual fluid will come out from vagina. That's why the opening should be partial. Okay. So these are the structure related to female reproductive, the external genitalia of female reproductive part. Okay. So now we'll try to observe the external genitalia. So this is the vulvar opening. This is the dorsal side, this is the ventral side. Sorry, opposite. This is the dorsal side, back side, and this is the ventral side, front side. Okay. So towards the ventral side, you will be finding urethral opening. Towards the dorsal side, you will be finding that vaginal opening. Okay. The outermost part, the fatty cushion with pubic hair, that is mons pubis. Below the mons pubis, you are getting labia majora. Below the labia majora, you are getting labia minora, right? And the upper junction, the ventral junction of labia minora, a finger-like structure is present, that is clitoris. And here you also will be getting the glands associated to female reproductive system, that is Bartholin glands and the paraurethral glands. So this is mons pubis, labia majora, labia minora, clitoris, this is urethral opening, this is vaginal opening. So that was all about female external genitalia. Okay. Now let's talk about a myth buster, a very common myth. Okay. So generally it's said that the hymen is often torn during the time of first coitus or first intercourse in normal time, right? So in old people, they used to tell that after marriage, if the hymen is intact, that means that is a sign of virginity. But that is not actual thing. So there are some other means the hymen can be torn. So what are they? However, it can also be broken by sudden fall or jolt. A sudden fall from cycle, from while you are dancing. Insertion of vaginal tampon. There are like few female, they will be inserting a cottony pad to absorb the excessive menstrual flow, that is called vaginal tampon. Okay, that will be inserted into the vagina. Active participation in some sports activities like horseback riding, cycling, swimming, dancing. In all the above cases, the woman may get a torn hymen. And even some women, after so much activities, even after coitus also, the hymen like is present in such a way it won't get broken or torn. So the torn hymen or intact hymen is not significant like measurement, either we are telling that a female is virgin or not. So in fact, the presence or absence of hymen is not reliable indicator of virginity of sexual experience. So that was the myth buster. Okay, now let's dive inside the like the very last topic of female reproductive system, that is mammary glands. Okay, so we know that mammary glands, they are responsible for lactation, milk production and milk secretion. So when we are talking about mammary glands, basically they are modified sweat gland. Mammary glands are modified sweat gland. Yes. The important question can be asked from mammary gland, that how it is made up of. The mammary gland is made by fatty tissue and alveolar tissue, which are responsible for production of milk. And every mammary gland is divided into many chambers, like 15 to 20 chambers, right? So one mammary gland is divided into 15 to 20 mammary lobes. There is one mammary gland that is divided into multiple chambers. That we are calling mammary lobes. Okay. Now what inside that mammary lobes? Inside the mammary lobe, you will be finding mammary alveoli. Okay, these are special structure which are responsible for production of milk. Okay. Now from mammary alveoli, some ducts will be coming out, and the ducts we are calling as mammary tubules. Many mammary tubules will join together to form mammary duct. Many mammary ducts will join together to form mammary ampulla or mammary ampulla. And from mammary ampulla, the milk will be sucked out through nipples, and the milk will be coming out from ampulla by lactiferous ducts. And ultimately will be sucked out through nipple. So you have to study again and again the passage of milk secretion or lactation. Again I am repeating it. If this is a single mammary gland that is divided into many chambers, we are calling as mammary lobes. Okay. Now inside each mammary lobes, what you are gonna get? Inside each mammary lobes, you are gonna get mammary alveoli. From mammary alveoli, the tubes which are coming out, that we are calling as mammary tubules. Many mammary tubules will join together to form mammary duct. Many mammary duct will join together to form mammary ampulla, and from ampulla the ducts are coming out, that is lactiferous duct, and ultimately milk will be sucked out through nipple. Okay. Now surrounded to nipple there is a dark zone, dark area, that we are calling as areola, and inside the mammary gland, that is mammary gland is supported with fat bodies. Okay, and behind the mammary gland you can find the section of ribs, the muscles between the ribs, the intercostal muscles, and obviously just behind the mammary gland the muscles are present. They are the pectoralis major muscle, right? So that was all about mammary gland. Few important thing when we are talking about the process of lactation, though we are gonna study in detail and in very short manner. Okay, I'm telling there is importance of two hormones. One is prolactin, one is oxytocin. There are two hormones which can influence the process of lactation. Prolactin responsible for milk formation and oxytocin is responsible for milk ejection. Now you are moving forward towards some important question related to this. So the following diagrams refers to the female reproductive system of human. Identify A to F. Okay, so this is A, this is B, this is C, this is D, and this is F, and this they are denoting as E. Okay, so obviously A is isthmus, B is ampulla, C is infundibulum, D is fallopian tube, E is ovary, F is uterine fundus. So option 4 will be our right match. Okay, now let's solve this question. Choose the correct option for given statements. Correct option. They are saying that female external genitalia do not include hymen and clitoris. Do not include. Correct statement you have to choose, right? So this is the incorrect statement. Mons pubis is a cushion of fatty tissue covered by skin and pubic hair in male. Again, incorrect one. The labia minora are paired folds of tissue under the labia majora. Absolutely correct. The opening of the vagina is completely covered by membrane called hymen. No, partially covered by. Completely, this is the wrong word. Okay. Next, the clitoris is a tiny finger-like structure which lies at the upper junction of the labia minora above the urethral opening. Absolutely correct line. Now they are saying that the hymen is often torn during the time of first coitus or intercourse. So we got that option 3 is correct, option 5 is correct, and option 6 is correct. So this is our right choice. Okay. Now let's come into another topic, that is gametogenesis. So the gametogenesis is divided into two sections. One is sperm formation or spermatogenesis, another is ovum formation or oogenesis. Okay. So we'll start with sperm formation or spermatogenesis. What is the location of this gametogenesis for sperm? If we are talking about spermatogenesis, then there is taking place inside seminiferous tubule. The highly coiled tubes present inside the testicular lobules, right? Okay. Now for spermatogenesis, the primary cells which we need, that is the germ cell or the spermatogonia. Now remember, they are diploid in nature, means each spermatogonia has 46 chromosomes. Now on puberty, when the process will start? The process will start on puberty, when we male are reaching into the adolescence, the age of adolescence. Okay. So spermatogonia will start division by the process of mitosis, will form more and more spermatogonia, and few of the spermatogonia will undergo differentiation, and they will get converted into primary spermatocyte. Primary spermatocyte, they are also diploid cells, means each primary spermatocyte containing 46 chromosomes, just like spermatogonia, just like they have a different shape in compared to spermatogonia. Okay. Now from spermatogonia, the actual story begins here. From primary spermatocyte, we will get secondary spermatocyte. From primary spermatocyte, we will get secondary spermatocyte by the process of a reductional division, that is meiosis 1. That means now if you observe into secondary spermatocyte, their haploid cell, they have only 23 chromosomes inside them. So if question is asked, which is the first haploid cell in the life of a sperm, then you have to answer secondary spermatocyte. They are not primary spermatocyte. First meiotic division takes place with primary spermatocyte and the result will be secondary spermatocyte. Now what after secondary spermatocyte? We will be getting spermatid cells and their result of meiosis 2, which is equational division. Okay. Meiosis 2 will result into spermatid, as because it was equational division, they have same number of chromosome, that is haploid, they have 23 chromosomes into them. So chromosome numbers are same like in secondary spermatocytes and spermatids. Okay. Now the cell division will be stopped here. Now what will happen? The spermatids, they need help from this Sertoli cell. They need help from this Sertoli cell. We know that Sertoli cells will be releasing a special hormone called sperm maturation factor. Now this sperm maturation factor will act on spermatids which are embedded into the lap or the villi or the microvilli lap of Sertoli cell or the nurse cell. They are continuously getting sperm maturation factor. Now they will start differentiation. No cell division will take place. Differentiation will occur under influence of sperm maturation factor, and the spermatids will get converted. The spermatids, they are spherical cells. They will get converted into tadpole-like swimming cells with n number of chromosomes, means with which are haploid nature and having 23 chromosomes into them, just like spermatids. So this was the structural maturation of the sperm, formation of spermatozoa. Okay. So throughout the process, if you observe it, the process we are calling it spermatogenesis. But if you observe only the last step, that is formation of spermatozoa from spermatid, this step is called spermiogenesis. Whole process spermatogenesis, only the last step of spermatogenesis, formation of spermatozoa or sperms from spermatids, that is called spermiogenesis, and also that is called structural maturation of sperm. And upon structural maturation, this mature sperm will be released out from the lap of Sertoli cell into the lumen of seminiferous tubule, and they will come out, and this process we are calling it spermiation. So there are three very important terminologies are there: spermatogenesis, spermiogenesis, spermiation. So what is spermatogenesis? Spermatogenesis is from spermatogonia. Through many sequential steps, we are getting ultimately sperm or spermatozoa. The whole process is called spermatogenesis. What is spermiogenesis? The very last stage of spermatogenesis, that is from spermatid to sperm or spermatozoa, that is spermiogenesis. And what about spermiation? Release of sperms or release of structurally matured sperms from Sertoli cells and into where they are getting released, into lumen of seminiferous tubules, and even when they are coming out of the seminiferous tubule, the whole process from Sertoli cells until unless they are getting matured, they will be embedded into the villi of Sertoli cell. When they are getting structural maturity, they will be released out into the seminiferous tubule. Now again, the peristalsis movement inside the tubules of testis, they will bring out the mature spermatozoa even outside. So this whole process we are calling it spermiation. So spermiation is a different thing, spermiogenesis is a different thing, spermatogenesis is another thing. Okay, so this diagram is taken from NCERT. So what this diagram is saying? So basically, this zone is towards the basal lamina of seminiferous tubule. We have taken a particular chunk inside the seminiferous tubule. These cuboidal cells, these are spermatogonial cells. Cuboidal or spherical, fewer cuboidal, fewer spherical. These are spermatogonial cells. While they will start division, they will form primary spermatocyte. After primary spermatocyte, we will get secondary spermatocyte. After secondary spermatocyte, we will get spermatids. After spermatids, we will be getting spermatozoa. Okay, now we will ask that where is Sertoli cell, sir? So basically, this is the nucleus of Sertoli cell, which is marked in NCERT, because this is the whole and soul elongated glandular cells or Sertoli cell. And the spermatids are embedded into their wall. See, now we will say that there are the spermatid cells and the sperm cells, they are similar as per this diagram. But remember, here if you observe them very minorly, you will find that the spermatids are larger, quite larger in compared to the sperm cells. So basically, when you are talking about spermatids, you will be finding two stages of spermatid. One is tailed spermatid, one is non-tailed spermatid. So the initial spermatid, initially the spermatids will look like a sphere, a football, and when they are going towards the maturity, they are just gonna form the spermatozoa. They will look like tadpole, or they have the flagella or the tails just like the sperm, but they are little bit larger in size. So the diagram is absolutely perfect, given in NCERT. Okay. Now again I am recapping the whole process. First we will get spermatogonia. Spermatogonia, they will divide by the process of meiosis to form more spermatogonia, right? By the process of mitosis to form more spermatogonia. Few of the spermatogonia, they will get enlarged, they will gain mass, and they will form primary spermatocyte. Now the primary spermatocyte, they will start meiotic division here. First there will be meiosis 1, which is reductional division. As a result, we will be getting from one primary spermatocyte, we will be getting two secondary spermatocytes. And both the secondary spermatocyte will perform meiosis 2 or second meiotic division, and the cells we are gonna get, they are spermatids. So who are haploid, who are diploid? Spermatogonia, haploid, primary spermatocyte, spermatogonia, they are diploid, primary spermatocyte, diploid. After first meiotic division, we got secondary spermatocyte, they are the first haploid cell. After meiosis 2, we are getting the spermatids, they are also haploid in nature. And these spermatids will undergo differentiation, they will just change their shape and will become mature sperm. We are calling them spermatozoa. So a very simple calculation can be asked, that how many sperms are produced from one primary spermatocyte. 4 sperms are produced from one primary spermatocyte. How many spermatids are produced from one primary spermatocyte? 4 spermatids are produced from one primary spermatocytes. How many secondary spermatocytes are produced from one primary spermatocyte, two secondary spermatocytes are produced from one primary spermatocyte. How many sperms are produced from one secondary spermatocyte? Two sperms and two spermatids are produced from one secondary spermatocyte. So as simple as that. So very simple calculative question can be asked from the process of gametogenesis. And when the process starts at puberty, and till your senescence or old age, it will be continuing. So it's a continuous process. Okay. So that was all about the process. Now what is the way the sperm will be coming out and how is gonna come out? So the way through which the sperms are coming out, it will be synthesized into the seminiferous tubule. After seminiferous tubule, they will get outside the seminiferous tubule and enter into the retitestis, and by retitestis it will enter into the vasa efferentia, vasa efferentia will proceed the sperms towards epididymis. Through epididymis. Now see, this is a temporary storage for sperm. What will happen into the epididymis? Functional maturity. Functional maturity of sperm will be happening here inside the epididymis, and inside seminiferous tubule structural maturity will take place inside the seminiferous tubule. Now what is the difference between functional and structural maturity? Once the sperms are getting the proper shape, there is the structural maturity, but it cannot move its tail or flagella. So that swimming movement we can observe just after the epididymis. That is called functional maturity of the sperms. Functionally they can swim now. So after epididymis, from the tail of epididymis, they will enter into the vas deferens. Now this vas deferens will get connection from seminal duct and will get ejaculatory duct. Ejaculatory duct will get connection from urethra, from prostate, and ultimately there will be formation of urinogenital duct or urethra, and it will be open through urethral meatus or urethral opening. Now let's study the structure of sperm. When sperms are structurally matured, we can divide the sperm into three parts: head, the neck part, the middle piece part, and obviously the tail part or the flagella of sperm. Okay. Now what are the structure you are getting in the head? In the head region, you are getting here acrosome at the edge and a condensed nucleus. Into the neck region, what are the structures we are getting here? you're getting two centrioles. Okay, one proximal and one distal centriole. The distal centriole is responsible for the formation of the flagellum or the tail part, okay, or the axial filament part. Who is forming it? The distal centriole, and the proximal centriole will be gifted to the ovum upon fertilization. So we can say that the centrioles which are present inside our cells, they are paternal, they are coming from father through sperm. Okay. Now if you look into the middle piece part, then you will find a spiral of mitochondria which are powerhouse, which will be utilizing the energy from the fructose, and it will be producing ATP for the propulsion or propagation of sperm or swimming of the sperm. Okay, so the energy we are getting from the mitochondria, the energy source for swimming, and the structure which is ultimately responsible for swimming, that is the flagellum or the tail of sperm. Okay. Now when we are talking about this acrosome of sperm, basically that is nothing but modified Golgi complex. That acrosome has many enzymes called acrosins or sperm lysins, which will be helping the sperm to penetrate inside the ovum during the time of fertilization and the fusion. Okay. The nucleus, that is haploid in nature, that is having 23 number of chromosomes. Okay, so that was all about the basic thing about the structural aspect of sperm. Okay, now some important talk about sperm. So if we talk about a healthy human male, that he will be ejaculating. Ejaculating means the way a male is releasing the sperm with a liquid medium, that altogether we are calling it semen. 200 to 300 millions of sperms during a coitus, which for normal fertility at least 60% of the sperms must have normal shape and size, and at least 40% of that 60% sperm should show vigorous motility, right? So see, for one ejaculation, a male releases 2 to 4 ml of semen. So semen means sperm plus the liquid medium which is laden with nutrition. Okay, that contains 200 to 300 million of spermatozoa or sperms. Now very important calculation is given here. If you are producing 100, suppose if you are producing 100 million sperm, then 60% of them, that means 60 million, should have normal shape and size, and 40% of this 60 million, that means 24 million sperms, should get vigorous motility, means high speed movement. If you get a question that the sperm count is 200 million, if the sperm count is 200 million, then how much sperm should have normal shape and size? So the answer will be 60% of 200 million, that is 120 million. And how much sperm should show vigorous motility, means high speed movement? That is near about 48 million sperm to get normal fertility. So that was about fertile male. Okay, remember always this 40% sperm with vigorous motility, that should be 40% of that 60%, 40% of not that total sperm or 100%, right? Okay. Now let's see what they are saying. Sperms are released from the seminiferous tubules and transported by the accessory ducts. Already we were studying this. And secretion of epididymis, vas deferens, seminal vesicles, prostate are essential for maturation. See, secretion of epididymis, they are responsible for the functional maturation, and secretion of other parts will help the sperm for its motility and survivability by providing them nourishment. Okay. Now let's talk about the semen. So what is semen? Semen is nothing but it has sperms along with seminal plasma. Now the seminal, what is seminal plasma? That is secretion of the accessory ducts and accessory glands. Okay. 70% of seminal plasma will be coming from seminal vesicles, 25% will be coming from prostate gland, 1 to 5% will be coming from the bulbourethral gland, right? So altogether the sperm and seminal plasma, we are naming it semen. Okay. Now let's come into the female part. So the process of formation of mature female gamete, that is ovum, is called oogenesis, which is markedly different from spermatogenesis. Now, how and where it's different? See, we know that the oogenesis process, it starts during the time of embryonic life. Okay. Starts during embryonic life of female. When it's not delivered yet, just it's in fetus condition, and when the age is near about 20th week, then the ovaries are matured and the ovaries will start the preparation of oogenesis. Okay. Now, what are the basic stages of oogenesis? See, first it will start with oogonia, which are diploid cell. Oogonia will get converted into primary oocyte, which are diploid again. Primary oocytes get converted into secondary oocytes. Which are first haploid cell of oogenesis, and secondary oocytes will get converted into oocyte, and that oocyte is also called ovum. Okay, so we need to study the process of conversion of this oogonia through all these steps, and ultimately formation of one oocyte or one ovum, okay, during the time of fertilization. Okay, so let's start the topic. Before this, we need to study the ovary in detail. Okay, so this diagram is directly taken from NCERT. See, listen like a story. So during the time of embryonic development, we'll get some diploid cells are forming inside the cortex of ovary. Those diploid cells, we are calling them oogonia cells. Right? Now, this oogonia, some of the oogonia, they will get converted into, primary oocyte by the process of mitosis and differentiation. They will just gain the mass. So those differentiated oogonia, we will be calling them primary oocyte. The differentiated oogonia, will be calling them primary oocyte. Here, to mention it shortcut manner, I will be just utilizing primary for 1 degree, secondary for 2 degree, tertiary for 3 degree. So the oogonia will be converted into primary oocyte, 1 degree oocyte, right? They are also diploid cell, they have 46 number of chromosome. Now this primary oocyte, they will start meiotic division or first meiotic division, but that will get arrested into prophase 1 of first meiotic division. Now why it's getting arrested? Now as soon as the primary oocytes are formed, after that we will find that some cortical cells or some particular cells, flat cells, are surrounding them. We call them follicular cells or granulosa cell. In the beginning we call them follicular cells, and after that we call them granulosa cells. So these granulosa cells or follicular cells, when they are covering, making one lining of covering outside the primary oocyte, they will secrete some inhibitory factor, and that will keep that primary oocyte arrested into prophase 1 of first meiotic division. And as soon as this primary oocytes that is encaged with the help of granulosa cell, we call that structure primary follicle. And how many primary follicles are produced here? Near about 2 millions. Primary follicles are produced here. Okay. Now they are waiting as arrested condition. Again, the process of meiosis 1 will resume during the time of their puberty, when that will get influence from the gonadotropin hormones, when the female will enter into the puberty. So within this long period of time, most of the primary follicle will die by the process of atresia. and ultimately during the time of puberty only 60 to 80,000 of those primary follicles will be surviving, and now the meiosis 1 will resume here and they will complete the process. Eventually the follicle will keep growing and the primary oocyte will get converted into secondary oocyte when the female will be reaching into the puberty. Every month one primary follicle will resume its journey towards the maturation, right? Now while these structures are developing, after primary follicle will be getting secondary follicle, after secondary follicle will be getting tertiary follicle. When the follicles are matured, we'll be getting Graafian follicle or the mature follicle, and upon stimulation of the hormone called luteinizing hormone, and also it will get help from FSH hormone and the estrogen, the Graafian follicle will get ruptured and it will throw out the secondary oocyte. And as soon as the Graafian follicle is emptied, the Graafian follicle will get converted gradually into a yellowish structure that is called corpus luteum. The yellow pigmented structure which containing the lutein pigment, that is basically the emptied Graafian follicle. So the yellow body or corpus luteum is formed, which will become a temporary endocrine gland that will become the main source of the hormone progesterone, and also it can produce estrogens as well. The corpus luteum will sustain for 12 to 13 days in normal female if there is no fertilization, and after that it will gradually start dying by forming a white colored body called corpus albicans. So let's observe the journey of ovum, right? So we are gonna study oogenesis in detail. So we are talking about embryonic life, and the cell here, that is oogonia. It is diploid cell, has 46 chromosomes. And the life is embryonic life. The oogonia will start division. After mitosis and differentiation, the oogonia will be forming 1-degree oocyte or primary oocyte, which is again diploid in nature. And how primary oocyte is formed from oogonia? As a result of mitosis and differentiation. Okay, that is in embryonic life. Now this primary oocyte will start for meiosis 1 or first meiotic division but it will be arrested into the prophase 1 and specifically the diploteny stage of prophase 1. That process is arrested now. So that was about the embryonic life. Now there will be the birth, childbirth. Now after birth, gradually the puberty arrives here. Now upon puberty, it will get stimulation from gonadotropins like LH and FSH. And the process will start resume again. So there will be completion of meiosis 1. So that primary oocyte will form two cells. One cell is quite larger, that we are naming the secondary oocyte, and one quite smaller cell, we are naming it first polar body. Now why is so? There is unequal cytokinesis. So this meiosis 1, it's completed, we can say that this is equal karyokinesis but unequal cytokinesis. And obviously the meiosis 1, there is a reductional division, there is not equational division. The nucleus, the chromosomal material will be divided. Half of the chromosome will go inside the secondary oocyte and half of the chromosome will go inside the first polar body. So secondary oocyte and first polar body, they both are haploid cell. They got equal number of chromosome in their nucleus, but the secondary oocyte in compared to first polar body got maximum volume of cytoplasm. That's why I am telling here unequal cytokinesis will take place here. Why unequal cytokinesis is taking place? Due to this long waiting of near about 12 to 13 years when the female will be reaching puberty, suppose it's 11 years or 12 years or 13 years. So due to this long gap, that cell, that primary oocyte will lose its centriole, and you know that importance of centriole during the time of cell division and during the time of cytokinesis, it maintain the cytoplasmic flow. So as because centriole is lost, there will be uneven division, unequal division of cytoplasm, and the cell which is getting more cytoplasm, that is called secondary oocyte. And what is the fate of the first polar body? You don't know. It may be die or it may be divide. NCERT is also not confirming about that. Now let's talk about the secondary oocyte here. Now eventually the secondary oocyte will be ovulated. The ovary, every month one single ovary will kick out the secondary oocyte from the Graafian follicle of the mature follicle, right? Now if the sperm is arriving, then the process will start again. So what happen after ovulation or during the time of ovulation, the secondary oocyte will start meiosis 2 or second meiotic division, but again get arrested into metaphase 2 of meiosis 2. So until unless the sperm is arriving or the fertilization is taking place, we won't get the oocyte or ovum. So in most of the cases, for every month of ovulation, they're just ovulating the secondary oocyte. In case of fertilization, only we'll be getting the ovum. So if sperm is arriving, the fertilization is taking place, then only the meiosis 2 will be completed, and upon completion of the meiosis 2, from the secondary oocyte, again we are going to get two cells, one large cell that is oocyte and one small cell that is second polar body, and both of them have haploid sets of chromosome, means 23 numbers of chromosome. So I am giving you one question here. Suppose I am giving you a question. There is one female, after marriage she has two children. She has done one abortion or one miscarriage was there, and her age is 38 years. The question is asked, in her life, how many ovum is produced? So you no need to calculate. Suppose also the data is given 13 years, this is the time when she started the process of menstruation, that was the menarche. So you no need to calculate each and every month. So the question is very easy. The question has asked that how many ovum is produced in case of that respected female. So you are getting two children, means twice the fertilization was there. One abortion means again the fertilization took place, right? So the answer will be thrice in her life. Thrice in her life she was able to conceive. So conceive means fertilization, though two are successful and one is unsuccessful, but we have to calculate that. So again the funda is similar, simple. If fertilization is taking place, secondary oocyte will complete second meiotic division and will form a single ovum. And if it's normal life, if there is no fertilization, the maturing ovum or the maturing egg will die in the form of secondary oocyte, right? Okay, so that was all about the process of oogenesis. Now the question can be asked that what is the difference between the marked difference between the spermatogenesis and oogenesis. So here you can tell that spermatogenesis is a continuous process, oogenesis is a discontinuous process. Twice in her life the process was arrested. First, during the time of first meiotic division, in the prophase 1, the primary oocyte was arrested, and secondly, after the secondary oocyte, just prior to formation of ovum, it will be arrested again into the metaphase 2 of meiosis 2. Only ovum will form upon fertilization. So it's a discontinuous process. Arrest, start, arrest, start, arrest. So twice you can see arrest is there, when the process is stopped there for further continuation. So there is a discontinuous process, oogenesis. Okay, the starting oogenesis will start during the time of fetal life or embryonic development. Spermatogenesis will start during the time of puberty. There is no prominent end point of spermatogenesis, but there is prominent end point of oogenesis, that is menopause. So these are the major differences. But what are the similarities? For both the cases, the first haploid cell will be here primary spermatocyte, here secondary spermatocyte, here secondary oocyte, the first haploid cell. Okay. But for in case of spermatogenesis, after spermatid formation there will be one differentiation and then we will be getting sperm. But in case of oogenesis, after formation of ootid, there is no further differentiation because that ootid only we are calling it ovum. So again, for oogenesis, you are getting only differentiation only once in their life when the oogonias are getting converted into primary oocyte. But in case of spermatogenesis, there are differentiation you can observe twice in their life. It is one spermatogonia to primary spermatocyte and another spermatid to spermatozoa, right? Okay. Now we need to conclude the topic with another important thing, that inside the ovary we may observe different stages, like starting from primary follicle, secondary follicle, tertiary follicle, Graafian follicle, then ovulation, then there is formation of yellow body, corpus luteum, and ultimately formation of the white body or corpus albicans. And inside them, in some cases, obviously we will be getting the developing stage of the egg, right? So inside the primary, so I am making one table. Here the stage of follicle, here the stage of egg. So the first follicular stage we will be getting the primary follicle, second stage is secondary follicle, third stage is tertiary follicle, fourth stage is Graafian follicle, and ultimately after Graafian follicle, when it's ovulated, we are calling it corpus luteum, and after corpus luteum is corpus albicans. So inside primary follicle we will be getting primary oocyte, inside secondary follicle we will be getting primary oocyte. Now on puberty, remember, on puberty the primary follicle will get converted into secondary follicle. Upon arrival of puberty, like before puberty, all the follicles are primary follicle. After puberty, every month primary follicle will start getting converted into secondary follicle to tertiary follicle. Okay. Now in tertiary follicle, you will find that the meiosis 1 is ultimately completed for the primary oocyte and you will get secondary oocyte. So first secondary oocyte is observed inside the tertiary follicle. Now the tertiary follicle will get maturity, the mature follicle is the Graafian follicle, and inside that Graafian follicle also you will find that secondary oocyte only, and ultimately that will be ovulated. Corpus luteum, there is no stage of egg you will be finding there. Corpus luteum, there is a temporary endocrine gland. And corpus albicans, basically It's white body, the dying corpus luteum. Corpus luteum, white's corpus luteum is the yellow colored body. Why it's looking yellow? Because of the presence of the lutein pigment, which is produced on the influence of the luteinizing hormone or LH, right? Okay. And how you can differentiate primary follicle to secondary follicle and secondary follicle to tertiary follicle? Primary follicle is having only few layers of granulosa cells. Secondary follicle, you may find one single theca layer. In tertiary follicle, you will find a fluid filled cavity called antrum and theca externa and theca interna. Two layers of theca you will be finding in tertiary follicle. Graafian follicle is much larger or much bigger than the tertiary follicle. If this is the primary oocyte and some granulosa cells are surrounding it, so this is your primary follicle. On puberty, that will get modification and you will be finding more granulosa layers surrounding it and outside a single theca layer. So this is the secondary follicle. The outermost layer is theca. The inner, they are granulosa layer. Here these cells are also granulosa. After secondary follicle, we are gonna get tertiary follicle. Tertiary follicle has a fluid filled cavity called antrum. This is the fluid filled cavity surrounded by granulosa cells, multiple layers of granulosa cell, and the theca has divided into theca externa, the outer theca, and theca interna, the inner theca. And when you will be getting an enlarged form, so this is the tertiary follicle. Theca externa, theca interna, this fluid filled cavity filled with the follicular fluid or liquor folliculi, that is called antrum. For the first time you can observe into a tertiary follicle. Okay. If someone asks you what is the importance of theca, see, theca externa, basically it's a fibrous layer. It's a fibrous layer and having some smooth muscle helps during the time of ovulation by creating pressure. Theca interna, that is basically the cells which will be producing some hormones. Basically the androgen hormones will be produced by theca interna upon the influence of gonadotropin hormones, okay, and the hormone will be converted by the granulosa cell into estrogens. So granulosa cells are also endocrine cells. Theca interna cells are also endocrine cells. Theca externa, basically they are fibrous cells along with some smooth muscle cell. Okay. So the largest follicle, it is the Graafian follicle, which is having a very large antrum, spelling remember, Graafian, Graafian follicle by the name of the scientist. This is the mature one. This is ultimately upon the influence of mainly LH hormone, and after ovulation it will release out the secondary oocyte, and it will get converted into a yellow colored body, corpus luteum, and the normal lifespan of the corpus luteum is 10 to 11 days, and after that the corpus luteum will die as corpus albicans. Okay. Now, when you can find the secondary oocyte inside the primary follicle, this is the primary oocyte. Secondary follicle, this is the again primary oocyte. The first secondary oocyte you will be finding inside the tertiary follicle, and Graafian follicle also has the secondary oocyte. And in Graafian follicle, outside the secondary oocyte, you will find a non-cellular layer, that is zona pellucida. A layer without any cell present outside the secondary oocyte, that is called zona pellucida. And when the secondary oocyte is ovulated, then if you study it, this is the haploid nucleus, this is the ooplasm, the cytoplasm of ovum is called ooplasm. Some tiny vesicles, granule filled vesicles you will be finding inside the cytoplasm, that is called cortical granules. They will be responsible for forming the fertilization membrane. After fertilization, the membrane of the egg cell, we are calling it vitelline membrane, or oolemma. Outside the vitelline membrane, the non-cellular layer or the acellular layer we are getting, that is called zona pellucida, that has receptor protein to recognize the human sperm. So the species specificity, zona pellucida layer is responsible for providing the ovum species specificity. And outside zona pellucida layer, the attached granulosa cells form a radiating pattern. We call them corona radiata. And the space between the vitelline membrane and zona pellucida is called perivitelline space. Now let's solve some question related to that. Statement 1 is saying LH acts on the Sertoli cell and stimulates secretion of some factors which help in the process of spermiogenesis. No, LH doesn't act on Sertoli cell. FSH will be acting on Sertoli cell. So that statement is wrong statement. Statement 2 is saying FSH acts at the Leydig cell. FSH acts at the Leydig cell and stimulate synthesis and secretion of androgen. No, again FSH will not be acting on Leydig cell, FSH will be acting on Sertoli cell. So both the statements are incorrect. Okay. Another assertion and reason, the assertion is telling the ovum retains most of the cytoplasmic content of the primary oocyte and much larger than the spermatozoa. Yes, ovum is much larger than the spermatozoa. The size of ovum is near about 100 micron and the size of the spermatozoa is near about 5 micron. So ovum is quite larger because after fertilization, the resource for the embryonic development you have to provide through your ovum. That's why it's necessary to retain most of the cytoplasmic content. And the reason is saying the ovum needs energy to activity to seek out the spermatozoa for the fertilization. Yes, both assertion and reason are correct and obviously they are the correct explanation. So we'll start with a new topic now, that is menstrual cycle, and with menstrual cycle we also have to study the hormonal regulation cycle in human female as well. So let's complete the next topic, that is menstrual cycle in human being. Okay. So now before starting the topic, I assume that many of you have queried that this is the reproductive cycle related to female, right? In female body, in every month is happening in female. By why it's named as mens? Okay, so the origin of the mens, the meaning of mens is month. That's because it's somehow similar to, that is 28 to 29 days, that is lunar month. That is the time a moon is taking to revolve around the earth. It is the lunar month. So that's why you are calling this like time period as menstrual cycle. So basically this is the reproductive cycle in female primates. Okay, now who are female primates? Are they all mammals? No. If you get a statement that all female mammals will undergo through this menstrual cycle, so that will be a wrong statement. That is exhibited by only female primates, and monkeys, apes like chimpanzees, gorillas and human being, these animals are considered as female primate. Okay, female of this particular animals. And as because it's a repetitive cycle, it's happening in every month, some sequential phenomena, some changes in ovary, some changes in uterus, and obviously some hormonal changes. So that's why the thing which is repeating again and again, we are naming it cycle. Now for human being, the first menstruation begins at puberty. Okay, what is puberty? The age of 11, 12 or 13 years. And we call the first menstruation as menarche. Even that is celebrated in many southern states in India. You know it very well. Okay. Now there will be permanent end. Wherever there is a precise beginning, there should be a permanent end of menstruation or reproductive cycle in case of human being, and that end is called menopause. Right? Pause means something has stopped. So the beginning is menarche, the ending is menopause. Okay. And we know the timing of the beginning. Now what is the timing of the ending of menstrual cycle or reproductive age of female? That is nearby 50 years of age. Around 50 years. Okay. Now it's saying that in human females, menstruation is repeated at an average interval about 28 to 29 days. So it's a repetitive phenomena. That's why we are calling it cycle. And the cycle of events starting from one menstruation to the consecutive of the next one is called the menstrual cycle. Now we'll study what are the sequential phenomena, what kind of changes we're gonna observe. Okay, so when you're talking about the menstrual cycle, there will be a precise ovarian cycle, the changes which are taking place inside the ovary, like there will be change in follicles. The primary follicle will start moving towards or start maturing towards secondary follicle, tertiary follicle, Graafian follicle, then ovulation, then formation of corpus luteum, corpus albicans. Then there will be uterine cycle, the changes which is occurring inside the uterus. Every month menstrual bleeding or uterine endometrium is getting sliding off or broken down. So again it's our body system is rebuilding it. So that's we calling it uterine cycle. So simultaneously we have to study the ovarian cycle and uterine cycle. Okay. Now all these changes will be influenced under the influence of varieties of hormones. So first set of hormones, that is anterior pituitary hormones, they're playing very crucial role. And what are they? They are follicle stimulating hormone FSH and LH or luteinizing hormone. Now primarily the follicle stimulating hormone, they are acting on ovarian follicles for the follicular development. That's why we are calling it follicle stimulating hormone. And upon the stimulus, ovarian follicle, specifically the cells of ovarian follicle, we are calling them granulosa cell, they are secreting estrogen hormone. And upon the influence of luteinizing hormone, the Graafian follicle after evolution will get converted into corpus luteum or yellow body, that is the primary source of progesterone. That's why if the question is asked, the secretion of progesterone is under influence of which hormone, you have to answer luteinizing hormone. Secretion of estrogen is under influence of which hormone? Primarily follicle stimulating hormone. Okay, right. Now these two hormones, obviously they are responsible for the secretion of some hormones from ovary, and those hormones are estrogens and progesterone. Now this estrogen and progesterone and their mechanism of secretion and action, their fluctuation in female blood will decide the uterine cycle. Now I am making a table where we are going to study what are the sequential phenomena we are going to observe. Ovarian cycle, uterine cycle. See, they are synchronizing with each other. Ovarian cycle and uterine cycle, they are synchronizing with each other. So the beginning of the cycle will be with menstruation and the bleeding of menstruation that is taking place inside the uterus. So we don't use any subsequent or synonymous terminology for the menstrual phase for ovarian cycle. Yes, but after menstruation, the phase will be calling as follicular phase or proliferative phase. Proliferative phase. Now the proliferative phase, when the uterine wall is getting proliferation after the menstruation again getting rebuild up, okay, renovation is happening. That's why we are calling it proliferative phase in terms of uterine cycle. Now simultaneously, while the proliferation is occurring into the uterus, inside the ovary, the follicles are getting matured. That's why you are calling this phase as follicular or pre-ovulatory phase. So they are synonymous. You can call the second phase of menstruation follicular phase, pre-ovulatory phase, or proliferative phase. Now what after proliferative phase? Now is the middle of the cycle. If the cycle is of 28 days, consisting 28 days, the very middle day, that is nearby 14th day, there will be ovulation. That is ovulatory phase, ovulation or ovulatory phase. Okay. Now for ovulatory phase, there is no synonymous terminology we will be utilizing for uterine cycle, because here also proliferation is going on. Now the last stage of uterine cycle and the longest stage of uterine cycle, that we call luteal phase. Luteal phase. The luteal phase, why? Because inside the ovary we can observe corpus luteum and the secretion of progesterone and estrogen hormone. So the luteal phase can also be called as secretory phase. Right? Now we'll be detailing each and every cell. Why the menstruation occurs? The absence of progesterone hormone, okay, stimulate the sliding of or breakdown of the uterine wall. There is menstruation. Which of the hormone or what is the hormonal relevancy with follicular phase or proliferative phase? Here the relevant hormone is for follicular phase for ovary FSH, and for proliferation of uterus, that is estrogen. So FSH and estrogen, the responsible for follicular phase or proliferative phase. For ovulation, the main hormone which is responsible, that is luteinizing hormone, as well as FSH and estrogen help the process. Okay. For luteal phase, the main hormone responsible, that is luteinizing hormone, for the initiation of the luteal phase or the secretory phase. Now once the corpus luteum is formed, now there will be production of progesterone and estrogen. Now the changes, subsequent changes, or the thickening and the maintenance of the proliferated uterus will be taken care by the hormones which are released by the corpus luteum. Okay, so let's study the process in detail. So the very first stage of menstrual cycle, if you observe the menstrual cycle like a clock, so from the very first day, if you start the ticking, the clock will end the ticking at the end of the one turn, that is on 28th day. So the clock is starting from first and ending into 28. So 28 days clock. So first, second, third, fourth and fifth day, the very initial 5 days of the cycle, we are naming these as menstrual phase or menstruation. What is the duration of the menstruation? From first to fifth day. Okay, sometime in some female it can be of 3 days, in some female it can be of 5 days. So in menstruation, what's happening? The uterine endometrium is getting broken and coming out in the form of bleeding. So that's why we are calling this process menstruation or menstrual bleeding. and that occurs due to the absence of progesterone, because the progesterone was the responsible hormone to maintain the uterine lining. Now, what after the menstruation? After the menstruation, we can observe the second phase of menstrual cycle, that is proliferative phase. Either you can call it proliferative phase or you can call it follicular phase. Now, what is the significance or importance of proliferative phase? Here you can observe the proliferation or the thickening of uterine endometrium is happening by the influence of mainly estrogen hormone. And from where this estrogen hormone is coming? The estrogen hormone is coming from the maturing follicle inside the ovary. So whatever things are happening inside the ovary, that is follicular maturation or follicular development, from primary to secondary follicle, tertiary follicle, then Graafian follicle. So this follicular maturation is named as follicular phase. So they are syncing with each other, proliferative phase and follicular phase. And this follicular maturation inside the ovary, they are under control of which hormone? They are under control of FSH hormone, follicle stimulating hormone. Okay. Now what after proliferative phase? That's near to the 14th day. And there is the middle of the cycle. That is the middle of the cycle. Here you can observe the name of the phase. Remember, that is ovulatory phase, or ovulation will occur under influence of mainly LH, and also FSH and estrogen helps in the they will be helping in the process. So from each from one of the two ovaries, either it's left ovary or right ovary, the secondary oocyte will be released out. So that's why we are calling this as ovulation. Okay. Now what after ovulation? The longest and the last phase of the menstrual cycle, and we call this as luteal phase. Okay, this is the longest phase. Now what's happening into the luteal phase? See, now the emptied Graafian follicle after ovulation, will get converted into a yellow colored body that is rich in lutein pigment, yellow color pigment. This is called corpus luteum. And this corpus luteum, they are becoming endocrine cell. They will soon start producing progesterone hormone, mainly in high amount, as well as estrogens. So as because they have started secretion of progesterone and estrogen, we also call the luteal phase as secretory phase. And in this phase, as because both the hormones progesterone and estrogen are present in the female blood in very high titer, high concentration, so the highest proliferation or the thickest lining of the uterine endometrium can be seen here. So that was all about the phases of menstrual cycle. Now we are going to come to the most conceptual part of menstrual cycle, that is a graphical representation is given in NCERT. So we are going to study that. Okay, so we'll start with first with the hormones of anterior pituitary. So we need to talk about two important hormones. First FSH, then it's LH. There the secretion will be influencing the menstruation or menstrual cycle. They are anterior pituitary hormones. This green line is, or the green graph is for the hormone, follicle stimulating hormone. And the white line is, that is luteinizing hormone. The white line is of luteinizing hormone or LH. So here it can be observed that nearby 14th day, both the hormones LH and FSH, they are reaching to their peak, and this phenomena is called as, when the jump of LH is there, that is LH surge, that is 10 times more amplification of the presence of this hormone into female blood, and this is FSH surge, two times to three times amplification of concentration of FSH in female blood, just the middle of the cycle, near to the 14th day, right? So you can observe that in the beginning and at the ending of the cycle, both the hormone, that is FSH or LH, at the low level, but at the center part, that is the middle of the cycle, both the hormones at their peak level, and LH is going towards a higher peak that we are calling LH surge, which is very much significant for the process of ovulation. Okay, now how these two hormones, the gonadotropins, we call them gonadotropins, right? They will be regulating the ovarian cycle or the follicular phases. How they will be determining? See, the follicular phase start with a small primary follicle. Then after primary follicle, you can observe the secondary follicle. After secondary follicle, you can observe a larger tertiary follicle. And the largest of among all the follicles, that is Graafian follicle, towards the middle of the cycle. Okay. And just near to the 14th day, what you can observe, when the Graafian follicle is getting ruptured out, and it's releasing out the secondary oocyte, is called ovulation. So in follicular phases, the first follicle, there is primary follicle or 1 degree, 2 degree follicle, secondary follicle, 3 degree follicle, tertiary follicle, and GF is not your GF, is Graafian follicle, is the largest follicle and the mature one. Okay. So here I must need to say that the follicular development is under control of this green hormone, that is follicle stimulating hormone. Follicle stimulating hormone is due to the influence of follicle stimulating hormone. the primary follicle is going towards maturation to form secondary, tertiary and Graafian follicle. Obviously, there will be influence of LH as well, but the main significant hormone here for the follicular maturation or follicular development, that is follicle stimulating hormone. And upon stimulation of follicular stimulating hormone, what you can observe here, you just you'll be observing secretion of estrogen hormone. Secretion of estrogen hormone. So if you observe the curve of estrogen secretion, so at the beginning the estrogen is near to 0. As soon as the follicles are getting developed, the estrogen, the curve of estrogen is rising. Follicles are getting bigger and bigger. They're gaining the granulosa cells and secretion of estrogen is increasing. So just at the middle of the cycle, you can observe the estrogen has reached into the peak of menstrual cycle. So the highest peak of estrogen is observed when there is Graafian follicle. That's because the follicles are under influence of follicle stimulating hormone, and upon influence of FSH, these follicles are releasing more and more estrogen with time. Okay. But one more thing here is very important. When the estrogen is rising, the level of estrogen is rising, okay, estrogen is reaching towards the peak, it will also send support. It will also send support to the gonadotropins hormone, that is FSH and LH, that is called positive feedback for their more and more secretion from anterior pituitary. So that is the reason just prior to ovulation or middle of the cycle, you can observe LH surged. See here, see the LH and FSH, they were increasing in normal manner. But why suddenly you can observe a surge of LH and FSH? Due to the positive feedback of increasing estrogen. So the gonadotropins and estrogen, they both are depending upon each other. They are helping each other for their secretion, right? Okay. Now you can observe that this LH, that is controlling Graafian follicle and helping the Graafian follicle for the ovulation and how it is taking place. Basically, LH is making the granulosa cells and the thicker cells to secrete more and more liquid inside that antrum, that is liquor folliculi. Now, when the liquor folliculi volume is gradually increasing, it's creating pressure from inside, and ultimately there will be a blast into that football of Graafian folliculi, and ultimately that force will throw out the secondary oocyte, and that we are calling ovulation. And as soon as the Graafian follicle is ovulated, it will get converted into a yellowish body which is bleeding from inside. So you may call it corpus hemorrhagicum, and that is a very temporary step, and that corpus hemorrhagicum will gradually get converted into corpus luteum, the yellow body. The maturation of corpus luteum will observe just near to 21st to 22nd day. The largest corpus luteum can be seen, and if there is no fertilization, so after that the corpus luteum again will start degeneration, and ultimately at the end of the cycle or the near to the 27th or 28th day, it will die by forming a white colored body, corpus albicans. Okay. Now what about the fate of estrogen here? Now initially, just prior to ovulation, the primary source of secretion of estrogen was this developing follicle. So after the ovulation, the follicles have gone. So that's why the estrogen has started deterioration. But as soon as the corpus luteum is developing, again there will be, you can observe, a rise in estrogen hormone. Okay. So again there will be second peak of estrogen whenever there is the largest corpus luteum is seen. So for a menstrual cycle, there will be two prominent spike or peak of estrogen can be seen. The first and the highest peak, that is due to the Graafian follicle, and the second peak is due to the secretion of estrogen from the corpus luteum. But along with this estrogen, the corpus luteum is secreting another important hormone, that is progesterone. So throughout the cycle, the progesterone level is very low. But as soon as the corpus luteum is forming, the progesterone level is increasing. And when the corpus luteum is mature enough, then again the first peak, the one and only peak of progesterone is seen here. But as soon as the corpus luteum is dying, the level of progesterone will deteriorate. So the yellow line is for progesterone graph and the orange line is for estrogen graph. Got it? Now we will observe how these two hormones, estrogen and progesterone, they are regulating uterine cycle. See, so in the beginning, what you can observe here, there is no progesterone, no estrogen. So particularly in the absence of progesterone, the uterine lining will start getting slouging off, that we call menstrual bleeding. And as soon as the estrogen has started to take off, the uterine thickness, uterine will start thickening its endometrium. And when it will enter into the luteal phase, again there will be rise of estrogen and obviously increasing titer of progesterone particularly. So that's why the estrogen will reach into the thickest thickest endometrium of estrogen is seen during the time of the middle phase of luteal phase, the middle zone of luteal phase. So from first to fifth day, first, second, third, fourth and fifth day, this span is for menstruation. From 6th day up to 13th day, this span is for proliferative phase. First is menstrual phase, 1st to 5th day, 6th to 13th day, proliferative phase, 14th day, ovulatory phase, and from 15th to 28th, that is luteal phase. At the middle of the luteal phase, you can observe the thickening of uterine endometrium is highest. And that is due to progesterone as well as estrogen. So if there is no progesterone and no estrogen, there will be no endometrium, and that's why we can observe menstrual bleeding. Now estrogen is increasing, uterine endometrium also start thickening itself. So who is responsible for proliferation of the cell division of the uterine endometrial cell? That is estrogen. Estrogen is responsible for making or thickening the uterine endometrium. Now here, why the thickest uterine endometrium is observed in the middle of the luteal phase is because you can observe progesterone level and estrogen level, both are at their high. So progesterone basically helps the uterine endometrium to maintain it, and the cells will be secreting nutritious fluid. So again I am repeating, estrogen is responsible for making and thickening the uterine wall. Progesterone is responsible for maintaining and calling more and more blood vessel to provide nourishment to the uterine wall. So the maintenance is the responsibility of progesterone, and making or building or thickening is the responsibility of estrogen. Now as soon as the corpus luteum gone, estrogen and progesterone is also gone. If there is no progesterone, as soon as the uterine endometrium will start getting sliding off, because blood vessel will die here. That's why progesterone is very much necessary to retain the uterine endometrium even during the time of pregnancy. That's why we call the progesterone as pregnancy hormone. Okay, now some common things about the menstrual cycle. See, the menstruation only occurs if the released ovum, or better to say secondary oocyte, is not fertilized. But if there is no menstruation or absence of menstruation, lack of menstruation may be indicative of pregnancy. Yes. See, every month menstruation is happening, but if a couple is doing regular coitus and in a certain month, if there is no menstruation, so that is the indicator the female partner is pregnant. However, it may also be caused due to some other underlying causes like stress, poor health, if someone is not taking care of the diet, not taking care of the hygiene, okay, or even in case of some diseases like PCOD, polycystic ovarian disorders, and some hormonal disbalance. In that case also irregular menstruation can take place. Okay. Now, during the pregnancy, all events of the menstrual cycle stop and there is no menstruation. Why so? During pregnancy is because during pregnancy the titer or the level of progesterone and estrogen is high. And a very much important word I am telling here. See, when alone estrogen is at its peak, then it will give a positive feedback to the gonadotropins. But if estrogen along with progesterone is going towards the peak, so both of the hormone is at their higher level, then they will give a negative feedback to the secretion, towards the secretion of gonadotropins. But during the time of pregnancy, there will be like higher level of progesterone and estrogen present in the female blood, and that will give a negative feedback to the female anterior pituitary. So that's why there is like no or very less amount of gonadotropins, that is FSH and LH will be secretion of FSH and LH will be seen. And if there is no FSH, no LH, all phenomena related to menstruation or menstrual cycle will be stopped. Okay. So that's why in the absence of fertilization, the corpus luteum degenerates. What happens? If fertilization is taking place, there will be formation of placenta to send the nutrition and the elimination of the harmful products from the embryo, right? So there will be a bridge between the embryo and mother body, mother uterus, that we call placenta, that start secreting a particular hormone called human chorionic gonadotropin, HCG, that is only produced upon pregnancy. So that helps the corpus luteum to retain its nature, to produce more and more progesterone during the time of pregnancy. But if there is no fertilization, the natural lifespan of corpus luteum or yellow body will get shrinked from 3 to 6 months to only 11 to 13 months. So in normal cases, the life of corpus luteum is 11 to 12 or 13 days. That is the luteal phase. Beyond that, it cannot survive. If you want to see your corpus luteum is surviving beyond that luteal phase, then you need to give it signal through human chorionic gonadotropin, which is only can be produced during the time of pregnancy. So that's why if the corpus luteum is degenerates, this causes disintegration of the endometrium and leading to menstruation because there is no LH, making a new cycle. And the cyclic menstruation is indicator of normal reproductive phase. Okay, if there is irregular menstruation, then there will be then that can be sign of tension, like something is happening inside the body. It may be related to the hormonal disorder, it may be related to the physical disorder. So normal reproductive phase means regular menstruation, and it extends starts from menarche and ends into menopause. This is the ending of the reproductive phase. And obviously you cannot forget the maintenance of hygiene and sanitation during the time of menstruation phase. Why so? See how it can be maintained. Female, they are using sanitary napkins or clean homemade pads, okay, during the time of bleeding, menstrual bleeding, for to absorb those things, right? Now you have to take care of the sanitary pads or sanitary napkins, that you have to change them within a duration of 4 to 5 hours. Remember it. Why so? After that, the sanitary pads or napkins will start through degeneration. Okay, and obviously there are certain chemicals which are present into the building material of those sanitary pads or napkins. They will also start leaking and they may harm your genital region. Okay, so you have to change them in regular interval, and when it's about the disposal of the used napkins, be precautious, be sensitive citizen, right? Because if you are throwing them here and there, they may cause soil pollution or water pollution, because there are certain harmful chemicals which you are gonna find inside those sanitary pads or marketed sanitary pads, which are really harmful for soil or aquatic bodies. So that's why be careful about maintaining proper hygiene and the disposal of those things. Okay, now a very important question is given here: why menstrual blood does not clot? See, if your menstrual blood is getting clotted inside your reproductive tract, then it will cause real problem for you, right? So that's why by nature the menstrual blood already have some anticoagulant mixed into them in the form of fibrinolysin and in the form of plasmins. They help the menstrual flow to blast the clot. They act as a clot blaster, and the fibrinolysin, that will be breaking down the fibrin, the fibers which are produced from the fibrin protein, and that will maintain the fluidity or the liquidity of the menstrual blood, so that it can be easily expelled out from female genital tract. Okay, now some questions. So first statement saying that rapid secretion of luteinizing hormone leading to its maximum level during the mid cycle, yes, near to 14th day, called the LH surge, yes, that induces the rupture of Graafian follicle and thereby the release of ovum, that we call ovulation. Instead of ovum, they should have written here secondary oocyte, but in normal term we can
assume that this is true statement. Okay. Statement 2 is saying that the corpus luteum secretes large amount of progesterone, which is essential for maintenance of the endometrium. Yes, corpus luteum, this is the primary source for progesterone. Also, it can secrete hormones like estrogen and relaxin or inhibin, but the main hormone released by corpus luteum is progesterone. And progesterone, without progesterone, you cannot maintain the uterine lining. If there is no progesterone into female blood, then immediately it will lead to the menstrual bleeding. So that's why, in the light of the above statement, choose the most appropriate answer. So both statement 1 and statement 2 are correct, but few of you may say that, sir, there should not be ovum, there should be secondary oocyte. Yes, that can be a concern, but in generalized manner, as because its ovulation is written here, we can consider statement 1 as a correct one. Okay. Now, the second question, the primary oocytes are in which stage of cell division? Okay. When the primary oocytes, the starting cell division, first they will start with meiosis 1. When? During the embryonic life of the fetus, of the female. Now suddenly they will get arrested. They will get arrested at which stage? The prophase 1 and particularly the diploteny phase, right? So our answer is prophase 1 of first meiotic division, right? Now let's complete the next topic. There is fertilization and implantation. Okay. So the fertilization can only occur if the ovum and sperms are transported simultaneously to the ampullary region. So that's why we call the fertilization is a chance factor. Fertilization is a chance factor. Okay, so that's the reason why not all copulations lead to fertilization. Very important line, statement based question and assertion and reason based question can be curated from here, right? Okay, so let's study the whole and soul process. Okay, suppose this is ovary, this is the uterine cavity, cervix and vagina, these are the fallopian tubes, ovaries. If there is ovulation, suppose from this particular ovary, the secondary oocyte will get inside the fallopian tube with the help of the embryo of the infundibulum part. And as because the secondary oocyte is immovable, the ciliary columnar ciliated epithelium, the columnar ciliated epithelium of the cilia will be helping the ovum to propagate through the infundibulum and to reach into the ampulla region, the site for fertilization, right? And similarly, the male partner has ejaculated the semen into the female vagina. Now, as soon as the semen is ejaculated, sperms start moving here and there. Now, the condition or the environment inside the vagina is acidic because the vagina inhabits some lactobacillus bacteria or lactic acid bacteria, right? So that's why due to the presence of lactic acid and those lactic acid bacteria or labs, vagina is generally acidic in nature, and the acidity is mandatory or the important to make the vagina infection free. Okay, so most of the sperms will die just after the ejaculation inside the vagina, what after that? As soon as the sperm will start dying, by seeing this casualty, the semen will clot itself with the help of some enzyme called clotting enzyme, which is already present inside the semen, right? Now, within 14 to 15 minutes, the clot will be declotted, and again, by crossing the cervical mucus, the sperms gradually enters into the uterus or uterine cavity. Okay. Now, what after that? Now, here you need a frantic head wash of the sperm. In the process, we are calling it capacitization. So, what is capacitization? Now, the sperms are moving really slow. Now, you need to speed up the sperm. Okay, if you want a successful fertilization. Now, this is the duty of the female reproductive tract. Now, the female reproductive tract will start releasing some fluid or chemicals which are responsible for this capacitization. Now, what is actually happening inside the capacitization? The acrosomal part of the head part of the sperm, okay, is laden with some cholesterol molecules, and during the time of capacitization, the cholesterol molecules will be wiped off from the sperm head, that is called head wash of the sperm, and suddenly the sperm will become more charged or energized. They will start rapid movement or rapid swimming. Now, while the sperms are swimming towards the fallopian tube, again, many of the sperm will die due to their abnormal size or shape. Some sperms are exhausted with their energy, and ultimately, some 100-200 sperms will manage to reach near to the ampullary region where the ovum is waiting. Now, the sperms can survive inside the female reproductive tract. The sperms can survive up to 3 days. The sperms can survive up to 3 days inside the female reproductive tract. Okay, at an average 48 to 78 hours, 72 hours, the sperms will be surviving inside the female reproductive tract. But the ovulated one, the secondary oocyte, it will be surviving only 12 to 24 hours maximum. Typical girly nature. It won't be waiting for the sperm. But like the guys or boys, the boys will be waiting for their female partner for a long time. Even few are waiting for whole life. Okay. A similar way, the sperm will show typical boy nature and the ovum will show typical girl nature. It will wait only for 12 to 24 hours. If the sperm arrives, then absolutely it will be fine for it. The secondary oocyte will be choosing the strongest sperm, and the chosen sperm will undergo fusion of the fertilization process with the secondary oocyte, and during the time of fertilization, eventually that secondary oocyte will get converted into ovum and oocyte, and ultimately upon completion of fertilization, what you're gonna get? We are gonna get a first unicellular embryo which is diploid in nature, that is zygote. Okay, now let's study the fertilization and implantation process. Now, how it happens? See, when the sperm is approaching towards the ovum, first it will get attached, it will make a temporary attachment with the zona pellucida. The lucky sperm is now attached with zona pellucida. What after that? Just after the attachment of the sperm with zona pellucida, now the secondary oocyte is agreed, yes, now this lucky sperm can enter through me. So, just after this magical touch, what will happen? Some changes will take place inside the ovum. Normally, the ovum is negative in terms of electrical charge or potential. So, there will be depolarization of ovum. There will be depolarization of ovum, and that we call fast block. That means that will block the other sperm to come closer near to the secondary oocyte. So from now the ovum is, or the secondary oocyte is very much loyal to the chosen sperm. So after the depolarization of the sperm, what will happen? The sperm will use its acrosome to penetrate inside the ovum. So there will be acrosomal reaction. The sperm, when it's getting penetrated into the ovum, complete sperm will enter inside the ovum, and the ovum, the secondary oocyte is now changing into Ootidor ovum, and it's securing itself that with the help of cortical reaction. It's securing itself with the help of cortical reaction. Now after cortical reaction, what will happen? Now it's completely secured. The ovum is completely secured. Now there will be intermingling with their cytoplasm, with their chromosome, with their nucleus, and a zygote will be formed. Now suppose this is the cell membrane of the ovum. The cell membrane is called vitelline membrane. Inside that, it has a haploid nucleus and some vesicles filled with cortical granules which are needed during the time of fertilization. Outside the vitelline membrane, a non-cellular layer is present which we call zona pellucida. Outside zona pellucida, some granulosa cells are there. They are called corona radiata. See, many sperms are trying to get that entry. But it's only the luckiest one will get the chance to get attached with the zona pellucida by forming a fertilization cone. Now, as soon as the sperm has formed a fertilization cone or touched the zona pellucida, the polarity inside the egg, which is negative, is gonna change into positive. It is called depolarization of the secondary oocyte. And when the egg has changed itself from negative to positive, that is called depolarization. Assume these sperms, these are also positive in nature. So except the lucky one, all the sperms will be getting a repulsion from the egg. They cannot even come close to the egg now. So that's the very quick process. That's why we are calling this process as fast block. So with this depolarization process, the secondary oocyte is actually blocking the other sperm to come closer. Right? Now what after that? The acrosome will release the acrosomal enzymes, and eventually the complete sperm will enter inside the complete sperm will enter inside the cytoplasm, and it will donate its haploid nucleus as well. Now while this process was going on, the ovum is produced from the secondary oocyte. So before this process, before the entry of the sperm, this entity we were naming it secondary oocyte. After the entry, we are calling it now oocyte or ovum. Okay. Now the cytoplasmic material is getting exchanged. Now their nucleus, gradually the nucleus will start a fusion, and ultimately that will form the zygote eventually. Okay. So what are the steps during the time of fertilization? The first step we will be calling it syngamy. What is syngamy? The magical touch of sperm and ovum. So when the sperm is getting penetrated into the ovum, so that process is called syngamy. Okay. Now within like some time, what will happen? They will start exchanging their, when the sperm completely got inside the ovum, what they will do? They will start exchanging the cytoplasmic material. So that is called plasmogamy. Now after that, what will happen? There will be fusion of the haploid nucleus of sperm and the ovum. The third step we call it karyogamy, fusion of nucleus. And obviously in the very fourth step, what will happen? The whole entity will get converted into a diploid zygote after the intermingling of the paternal and maternal chromosome, and that process we are calling it amphimixis. So what is amphimixis? Intermingling or intermixing of the chromosomes of sperm and ovum. Syngamy, the magical touch of sperm and ovum. Plasmogamy, exchange of the cytoplasmic material. Karyogamy, fusion or nuclear fusion. Amphimixis, intermixing of the chromosomes. So that's how the process of fertilization was done. Now we have arrived towards a zygote. Now before talking about the zygotic development, while the zygote was forming, we need to talk about how the sex is determined, that is sex determination. Now it's often said that the female partner is responsible for the sex determination, but it's actually the father. How? See, for the male partner, what is the chromosomal setup? 44 plus XY. For the female partner, what is the chromosomal setup? 44 plus XX. 44, the number of somatic chromosome or the autosome, and XY or XX, they are the allosome or sex chromosome. Now while male will be forming gametes, it will form two kind of sperm. With one sperm you will get 23 chromosomes with 22 autosome and the X allosome, and the other sperm with 23 chromosome will get 22 autosome with the Y allosome. So this sperm is called X sperm, this sperm is called Y sperm, right? Now what about female? Female can only produce ovum with 23 chromosomes, 22 autosome, and only X allosome or sex chromosome. So that's why female, they are called homogametic, male they are called heterogametic. Now wherever there is fusion between X sperm and X ovum, there will be formation of a female baby, female child, 44 plus XX. When there is fusion of the Y sperm and X ovum, there will be formation of male child or male baby. So the percentage or the probability of formation of male and female is 50%. So it's not upon the female who can choose, it's upon the male sperm that between the X and Y sperm, which of the sperm gonna fuse with the ovum, because ovum is always X. That's why the process of sex determination or the sex is determined by the male partner, okay, not by the female partner. Okay, now what are the steps of fertilization? See, now what will happen? The zygote, the first diploid cell, now it will start division. The zygote will start division. So the zygotic division will call cleavage. We will not be calling them mitosis, though they are technically mitosis kind of cell division, but we will be calling them as cleavage. Now why we call the division as cleavage? Because when you observe the zygotic division, suppose this is the zygote, and it will divide from one cell to two cell. Gradually, the cell will start decreasing their size. The size of zygote is not increasing. The size of the whole structure is not increasing. So the size of cells are decreasing with every division. So zygote, this is one cell stage. After that, there will be two cells stage of the embryo. After that, there will be 4 cell stage of the embryo, right? So with every division, the cell size, the number is increasing, and even these cells are carrying the similar number of chromosome which is present in the zygote. All the cells are diploid in nature only. So technically they are mitosis. But what happens in mitosis, if this is the mother cell and if two daughter cells are formed, they are of similar size. So in normal mitosis, the mother cell and daughter cell are of similar size. But in case of cleavage, the chromosomal number will be similar like mitosis. Chromosome number will be similar like mitosis. So in mitosis also we can observe that twin daughter cells are forming from twin mother cell. Here also similar thing, but cells are getting decreased in their size. Why it's so? Because the zygote was covered by zona pellucida. After first cleavage or first division, even the structure is surrounded by the zona pellucida. The second cleavage, even the structure is again surrounded by the zona pellucida. So that's why on sequential divisional phase, which is because the entity is confined by volume due to the presence of zona pellucida, which is a very hard layer. The cells are not getting space to increase their size for the division. Okay, so in that case, what will happen? The cell will decrease their size to to increase their number. So that is the difference between cleavage and normal mitosis or mitotic division. So after first cleavage, zygote will form a 2-cell stage embryo. Second cleavage, it will form 4-cell stage embryo. Third cleavage, it will form 8-cell stage embryo. After that, it will form 16-cell stage embryo. After that, it will form 32-cell stage embryo. So all of these are sequential stages of the embryo, and the very initial cells which are present inside that embryo, all of these are embryonic developmental stages. The cells are called blastomeres. The initial stages, 2-cell stage, 4-cell stage, 8-cell stage, so each and every cells of initial human embryo or initial embryo, will be calling them blastomeres cells, and they are totipotent or pluripotent in nature because they have the capacity to regenerate whole human body, or they have the capacity to produce organs and systems. Okay, they are with high potency. They are also acting as stem cell. Okay, so 2-cell stage, 4-cell stage, 8-cell stage, 16-cell stage, 32-cell stage, just like that the embryo will keep increasing the cell number, and here 8 to 16 cell stage we are calling it as morula. Okay, and beyond that, after that, the cells will be calling at the stage we will be calling at blastocyst. Okay, so here in the diagram you can observe this is the zygote which is confined inside the zona pellucida. Okay, this is the secondary oocyte. After fertilization, it is converted into diploid cell zygote. After first cleavage, we are getting 2-cell stage, and these 2 cells are surrounded by zona pellucida. After second cleavage, we are getting a 4-cell stage. Again, these are surrounded by zona pellucida. After that, we are getting 8 to 16 cell stage. It is again surrounded by zona pellucida. And as because this looks like a bunch of mulberry fruits, we call this stage as morula. Now, after morula, what? After morula, we can observe the stage which is a hollow football like structure, which is called blastocyst. And if you observe the blastocyst, there are 2 layers of cells present. Few cells present outwards, and few cells which are present inwards. So in blastocyst, the cells which are present outwards, we call them trophoblast. And the cells which are present inward, we call them inner cell mass. Now, this is the time that blastocyst is coming downwards from the fallopian tube. Even the morula, actually morula, which is coming downwards from fallopian tube to the uterine cavity, and it will get converted into blastocyst. And as soon as the blastocyst will digest the outer zona pellucida layer, the trophoblast cell will be releasing some zona pellucida digesting enzyme, and as soon as that zona pellucida is digested, the blastocyst will get embedded into the uterine endometrium, and that process we are calling it implantation. So which stage of embryo is getting implanted here? Blastocyst. Blastocyst stage of embryo is getting implanted here. See what happened. This is the early blastocyst stage. The outer layer of cells. Which we are calling trophoblast or trophoectoderm, and the inner cells we are naming them inner cell mass, ICM, inner cell mass. Now until unless there is zona pellucida, there won't be implantation. So why there is zona, these embryos are stages of embryos are carrying zona pellucida, is helping the embryo to decide when and where to get implanted. If there is implantation of embryo in early stage, that may lead to implantation in wrong place of uterus, and that we call ectopic pregnancy. See what happens if you observe the uterine cavity. There should be implantation either into the uterine fundus zone, or there should be implantation into the lower part, not too much lower, just near to the mid-ventral line of the lateral walls of uterus. Either this is a good place for implantation, or this can be a good place for implantation, or else this can be a good place for implantation. What are the wrong sites of implantation? This is the wrong site of implantation. Towards the cervix, that is the wrong site of implantation. Even sometimes if implantation is taking place inside the fallopian tube, that is also wrong site for implantation. So the zona pellucida is determining the proper time and place of implantation. Now what happens? The trophoblast cell, generally they will start secretion of some enzyme, and zona pellucida get digested, and the blastocyst, which is having a hollow empty space, we call blastocele, will come out from the zona pellucida coating Now, some of the trophoblast cells, they will be forming villi. And with the help of this villi, this blastocyst or the late blastocyst gonna embed itself inside the uterine endometrium, and soon it's got attachment with the uterine endometrium. The uterine lining will cover it. The uterine lining will cover it. So this was the process of implantation. Now, which are the cells helping into implantation? Trophoblasts. They help in implantation. The inner cell mass, they act as stem cells. Inner cell mass, they act as stem cell, and from here soon we are gonna get the baby or the fetus or the embryo. The body of that fetus or the embryo we are gonna get from inner cell mass, the cells which are present inside the blastocell cavity. Okay, so here a detailed diagram after the implantation of human embryo is given. So you can observe from here that how the embryo is implanted inside the uterine endometrial layer, the innermost layer of uterine endometrius, and as soon as is implanted, the mother tissue or the uterine tissue will also form interdigitation with the chorionic villi, or the villi, the structures which are responsible for the attachment by puncturing the uterine endometrial cell that we call chorionic villi. Along with the chorionic villi, the mother cells or the uterine cells will form a disc-like structure here. A disc-like structure here, which act as the bridge or passage of nutrients and the passage of exchange of excreta, elimination passage of excreta, and this common feto-maternal tissue we are calling it placenta. We call this feto-maternal tissue as placenta, right? Okay. So now immediately after the implantation, the inner cell mass of the embryo differentiate into the outer layer called ectoderm and the inner layer called endoderm. So just after the implantation, now the blastocyst is gonna get transferred into another next phase of the embryonic development that we call gastrula stage. Now what are the stages of gastrulation? First, see, you need to form a triploblastic body. So first the ectoderm and endoderms are formed, and after that the appearance of mesoderm will be there in between ectoderm and endoderm. So that is called trilaminar disc. Now, okay, so again a very important statement based question can be asked. First, during the time of embryonic development after the implantation, the blastocyst will get converted into stage gastrula. So in gastrula stage, what you can observe, first there is formation of ectoderm and endoderm, and after that in between them there will be formation of mesoderm. So from here the triploblastic development process will start. So these three layers give rise to all tissues or organs in adults. So that's why the ectoderm, mesoderm and endoderm, these are called embryonic germ layers. Embryonic germ layers. Now it needs to be mentioned here that the inner cell mass contains certain cells called stem cells. So where you will be finding stem cells? The cells which are responsible formation of the responsible for the formation of the organs and the body. Stem cells which are present in the inner cell mass, not in the trophoblast. The cells of trophoblast, they are responsible for the implantation and formation of the placenta. Yes. So which are the potency? Now I'm talking about the stem cells here. The stem cells, they have the potency to give rise to all the tissues and organs. So you can call them pluripotent stem cell. If a cell is having capacity or potency to recreate a full body, that is called totipotent stem cell. So zygote is a totipotent stem cell, or even the initial cells that we call blastomeres, they are also totipotent stem cell. And the cells of inner cell mass, they can be considered as pluripotent stem cell because they have the capacity to produce most of the organs and systems and tissues. Okay. So that was about the embryonic, the starting of the embryonic development.
So my dear students, so far we have studied the process of gametogenesis, the process of insemination, and after the insemination we have studied in detail the process of the fertilization and the implantation of the blastocyst stage of embryo into the uterine endometrium and the subsequent stages, right? So now let's talk about, like, after the placentation or the formation of placenta, which is a bridge or feto-maternal tissue in between the fetus and mother body, what is the significance of that particular structure? So we are going to talk about placenta. So placenta is basically a feto-maternal tissue. Now what do you mean by feto-maternal tissue? To form placenta, you need this chorionic villi, and this chorionic villi, they are structure originated from the trophoblast cell, and the outermost covering of the fetus, the developing embryo, that we call chorion. The outermost covering of the developing fetus, we will be calling it chorion, which is one of the extra embryonic membrane or extra embryonic covering. And along with that, the tissue from mother will be forming interdigitation. Interdigitation, like, means this kind of structure to hold that embryo properly, okay? And there, this formation of disc, which we are calling feto-maternal, some part is from fetus, some part of the structure is from maternal body or mother. Now what it will be doing? Transportation or delivery of essential nutrients, exchange of gases, between the developing fetus and mother body, and removal of excreta. Removal of excreta from the fetus. Remember, before birth, circulatory system will start working, excretory system will start working, but the respiratory system in the lungs will not start working. Lungs will start their work just after the birth, which you call switchover process. We'll study into the process of parturition. So basically, placenta is the bridge between, like, it's providing the useful material to the fetus and eliminating the harmful material from the fetus. So that is the importance of placenta. As well as the placenta act as endocrine gland. Now we'll talk about the endocrine function of placenta. So what are the hormones produced by placenta? So let's next topic we're going to study the hormones of placenta and what are their importance. Okay, so the placental hormones are obviously the most important one, HCG, human chorionic gonadotropin. Now, what is the function of HCG, human chorionic gonadotropin? Just after the placentation, it starts producing HCG, and which will be giving signal to the corpus luteum of the ovary, which was intending to die after 11 to 12 days. So, when it's getting the signal in the form of HCG that yes, implantation took place, fertilization successfully occurred inside that female body, now suddenly the corpus luteum will it will gain some more energy, recharge, it is just recharged, so that it will sustain for further 6 to 8 months. Okay, because corpus luteum, what is the normal lifespan of corpus luteum? 11 to 12 days. In the absence of fertilization, it will die, right? But whenever there is fertilization and implantation, placenta will start producing HCG, and that HCG will give that good news to the corpus luteum, and corpus luteum will survive for at least next 6 to 8 months. Okay. Apart from HCG, there is another hormone, human placental lactogen, HPL, that is necessary for the maintenance of the pregnancy, and also that helps the mammary gland of mother for its proper developmental process. Human placental lactogen. Okay. Another hormone of placenta is relaxin, though the relaxin is also again produced by the ovary, that is from corpus luteum. The relaxin hormone that is necessary particularly just prior to the parturition. The relaxin helps the cervical canal, the birth canal, to dilate. The relaxation of the birth canal just prior to the expulsion of the baby during the time of parturition. So, relaxin is an important hormone required during the time of parturition. Okay. Apart from these hormones, placenta produces estrogen, placenta produces progesterone, estrogens and progesterone. Placenta produces corticotropin releasing hormone, CRH. In normal human being, this is also produced by our hypothalamus, CRH, corticotropin releasing hormone. But here during the time of pregnancy, placenta also will be producing corticotropin releasing hormone. Which is again a very necessary hormone to induce the process of parturition. Okay. The ratio of estrogen and progesterone, the high ratio of estrogen and progesterone is mandatory to maintain the pregnancy, and when the pregnancy will be terminated by the process of parturition, then you need high level of estrogen and low level of progesterone. Remember that. throughout the pregnancy you need high level of estrogen as well as high level of progesterone. But when you are going for parturition, the baby delivery, then you need high level of estrogen but low level of progesterone. So maintaining these different ratios in different time duration, different timing is very much important for maintaining pregnancy and terminating the pregnancy. So these all are placental hormones. Okay. Now a question is asked that 3 hormones which are exclusively produced during the time of pregnancy. So what are those 3 hormones? They are HCG, human chorionic gonadotropin, they are HPL, human placental lactogen, and relaxin. HCG and HPL, they are produced by placenta. Relaxin, this hormone is produced by placenta as well. In NCERT it is given that the source of relaxin is also ovary, that is from corpus luteum of ovary, right? So relaxin can be produced from both the placenta and corpus luteum of ovary, and these 3 are exclusive. These 3 hormones will be finding during the time of gestation or pregnancy, right? Okay. One more question is asked that apart from this hormone, like what all other hormones also, there are few other hormones which are required to maintain the metabolic process and the whole and sole purpose of the pregnancy. So apart from these hormones, we need high level of thyroid hormones, thyroxines. We need high level of cortisol, that is the stress hormone. Now just after the parturition, you need to prepare the mother, specifically the mammary gland, to produce milk. That's why you need to maintain high level of prolactin. Okay, so during the time of pregnancy, you need to maintain high level of thyroxine or thyroid hormones, cortisol hormone, which is acting as a stress hormone, also help during the time of parturition. Okay, prolactin, which is necessary for milk production, as well as along with the hormones like estrogens and progesterone. These all of these hormones you require to maintain at high level during the gestation period or during the pregnancy. Right? Okay. Now we'll move forward to the next topic, that is the different stages of embryonic development. Now before going into that topic, just we'll do a quick recapitulation whatever we have studied in terms of the process of fertilization and embryonic development. So when we are talking about the process of fertilization, this is the fusion of sperm and ovum, fusion of sperm and the secondary oocyte. Basically before fusion it is not ovum, that is secondary oocyte. Okay. Now, during the time when the sperm is getting entry inside that secondary oocyte, that will get converted into ovum from secondary oocyte, and there will be formation of fertilization membrane, and once the fertilization process is completed, we'll be getting a single cell with 46 chromosomes. We are calling it zygote. Now the zygote will start its cell division inside, that we call process of cleavage, right? Now what is the difference between cleavage and mitosis? In cleavage the cell size will be increasing with every division, but in mitosis the cell size remains same. In cleavage, the new cells which are forming from the old cell, we will be calling them blastomeres. In mitosis, the new cells are called as daughter cell. Okay. Now, why in the process of cleavage the cells are not gaining the size, or the cells are decreasing their size? That is because in mitosis, in the interphase, there will be G1 phase, S phase and G2 phase, that is growth phases are present. But in case of the interphase of the cleavage division, only the synthesis phase or S phase is present. G1 and G2 phase are very negligible, or they are absent. So the main difference is their size. Mitosis, the cell size along with the chromosome number is maintained, but in case of cleavage, chromosome numbers are maintained, but cell size get deteriorated or decreased. Okay. Now after zygote, we will be getting the embryonic stages. When the embryo is 8 to 16 cell stage or 8 to 16 blastomere stage, we call that embryonic stage as morula. Now the morula will get developed into a hollow football like stage, that is called blastocyst. Now that blastocyst will get implanted. Implantation of blastocyst will take place, and after that we will be calling that embryo as conceptus, and that conceptus is prevailing in which stage of the embryonic development? That is gastrula. Now in gastrula, for the very first time you can observe first ectoderm and endoderm, then mesoderm. So from here triploblastic development will start from those embryonic germ layers, ectoderm, mesoderm, endoderm. Gradually there will be formation of all type of tissue, like first there will be epithelial tissue formation, then there will be connective tissue formation, then there will be muscular tissue formation, then there will be neural tissue formation. Okay. So gradually the gastrula will go further for the embryonic development, and soon the organ formation will take place, that is organogenesis or the morphogenesis process. Now let us study about the process of morphogenesis. The process of morphogenesis. Okay. So in NCERT, the process of morphogenesis that we need to study for 9 months, that is the gestation period for human being, and also questions are asked that in NCERT itself, what will be the gestation period in case of dogs, cats and elephants. So I am giving the answer. For dogs and cats, the gestation period is nearby 2 months, 63 to 65 days, and for elephants the gestation period is nearly of 2 years, that varies from 18 months to 22 months in case of elephants. Okay. For human being, the gestation period of the pregnancy duration is 9 months. Okay. Though in higher classes you get to know that it may vary from 7 months to 9 months. Okay. Now these 9 months, they are divided into 3 terms. First 3 months, the first trimester, Now, first, second, third month, first trimester, fourth, fifth and sixth month, we call these 3 months as second trimester. Seventh, eighth and ninth month, we call this the terminal 3 months as third trimester. So, trimester wise we need to study the organogenesis or the morphogenesis process, right. So, in NCERT, they have told about the developmental process in the first month, developmental process in the second month, in third month, fifth month, sixth month and ninth month. The important phenomena are described in the NCERT. Okay. So, the first month, what we can observe? See, we have one heart, remember, we have one heart. So, that is why that is related to first month, the formation of heart and first heart sound. So, in first month, what we can observe? Formation of heart, and also you can hear the first sound of heart by using stethoscope, right. But if someone asks you what is the first system which is getting developed into that fetus, then your answer should not be circulatory system, then your answer should be this neural system. Neural system is the first system which is getting developed, but when you are talking about the organ, then for the very first month we can observe the formation of heart. Okay. Now, what about the second month? See, we have 2 hands and 2 legs. So, in this way you can remember we have 2 hands. So, the hand formation, that means formation of the limbs, that is 4 limb hand and legs are hind limbs. Formation of limbs and also digits can be observed during the time of second month. Formation of limbs and digits observed during the time of second month. Okay, right. Now come to the third month. Now, how many genders are present in human being? 3 genders, like male, female and the transgender. We have 3 genders. So, the formation of genders can be like recognized by observing the genital organs. So, formation of genital organs and most of the important systems you can observe during the time of the third month. So, 3 gender, third month. Formation of genital organs and important systems or organs. Now, let us enter into the second trimester. Third month, that is the end of first trimester. Now, what is going on in fourth month is not given in NCERT. Now, directly we will be jumping into the fifth month. So, in fifth month, you can observe the first movement. The fetus is saying hi, you are saying bye. No, how many fingers are present? 5. Saying hi. First movement, F for first, F for fifth. So, in fifth month, you can observe the first movement of fetus and it is doing like this. Imagine, it is just doing like this. So, that means formation of hair on head is also observed in during the time of fifth month. First movement of fetus. and formation of hair on head. Now let's come to the 6th month, that is 6th month, that is the end of the second trimester. Now we have how many eyes? We have 2 eyes. So 2 eyes, second trimester, and from here the fetus will start blinking. Now how you can blink? Once your eyelids are separated. So in 6th month you can see the eyelids of fetus are separated, and also occurrence of eyelash, and body is also covered by fine hair. Okay, so formation of eyelash, eyelid separation on the second trimester on the 6th month, and by the time of the ending of the 9th month, that is 3rd trimester, the fetus is ready for parturition. Okay, so in diagrammatic format I have elaborated here. So if the question is asked, what are the major features of embryonic development at various months of the pregnancy? So the formation of heart, it can be observed to the very first month. Here you can observe, you can listen heart sound by using a stethoscope. Okay. Now what about the second month? Second month you can observe formation of limbs and digits, formation of limbs and digits, 2 hand, 2 legs. So limbs and digits formation on second month. Now where you are coming to the 3rd month. 3rd month you can observe the formation of external genitalia. Now by observing the fetus, the fetal image, doctor can determine either the fetus is male, female. So genital organs or external genitalia is formed during the time of 3rd month, along with most of the important organ systems. Okay. Now by the time of 6th month, or 2nd trimester, the eyelids are separated along with formation of eyelash, and body is also covered by fine hair, right? Okay. Next, what you can observe during the time of 9th month, the fetus or the baby is ready for the childbirth, the delivery. Next, the process of parturition. Remember this parturition, that is a complex neuroendocrine phenomena that will be regulated by signals of neural system as well as endocrine system. So the average duration of human pregnancy is about 9 months, which is called the gestation period, or in normal language pregnancy period. Vigorous contraction of the uterus at the end of the pregnancy causes the expulsion or the delivery of the fetus, that in normal language we call it labor. That vigorous contraction and the discomfort and pain which is occurring. The process of delivery of the fetus is called parturition or childbirth. Now the question is asked here that parturition is induced by a complex mechanism. What kind of complex mechanism? Complex neuroendocrine mechanism. Okay, now let's study that complex neuroendocrine mechanism. Now, very important question is asked that what are the signals or what are the factors which will be stimulating the mother's body to initiate the process of labor? That will be coming from the fully developed fetus and placenta. Fully developed fetus and placenta, they will generate a signal, a complex neuroendocrine signal that we are calling fetal ejection reflex. Fetal ejection reflex. That fetal ejection reflex induces mild contraction of uterus or uterine muscles which are present in the myometrium. You know, the contraction of uterine myometrium is very much necessary for the process of expulsion or the parturition of the baby. So, when the fetus and placenta, they are fully developed, they will generate a reflex that we are calling fetal ejection reflex, that will introduce induce, means stimulate those myometrial muscles to perform a very light contraction, that is called mild contraction of uterine muscle, and after that what you can observe, that will stimulate, that will send a positive feedback to maternal pituitary. And the posterior part of maternal pituitary, that is a reservoir of which hormone? ADH hormone as well as oxytocin hormone, which is necessary. Now, this maternal pituitary will release, actually the oxytocin is produced by hypothalamus, but will be released through the posterior pituitary. The release of oxytocin. Okay. Now that oxytocin will come and bind with the receptor of uterine muscle. Now remember that to promote the formation of oxytocin receptor on uterine muscle, you need to maintain high concentration of estrogen and low concentration of progesterone here. That's why we can see that drastic fall in the concentration of progesterone and rise in the concentration of estrogen, the terminal phases of the embryonic development. So, oxytocin is the friend of estrogen is the friend of oxytocin, and progesterone is the enemy or foe of oxytocin. That's why under influence of estrogen, the oxytocin will react more. Release of oxytocin, that will increase uterine contraction, and ultimately that will cause expulsion of fetus, that we are calling the childbirth. Okay. Now, while this expulsion of fetus, the process was going on in the background, few more hormones were acting on uterus. You cannot overlook them. They are prostaglandin along with oxytocin. they help in uterine contraction. Relaxin, one of the most important hormone which is needed during the time of this process of parturition. Relaxin, what it will do? Relaxin will help to dilate, to widen the birth canal by giving signal, by relaxing the muscles of the cervix, right? Okay. So, relaxin, prostaglandin and oxytocin, they are very much necessary during the time of parturition process, and sometime if naturally mother body is unable to fail to produce good amount of oxytocin, then doctors will be injecting the synthetic oxytocin, that is called pitocin. So, in NCERT it is asked that sometime doctors will be giving injection to the mother, that is actually oxytocin, in the name you will be getting the marker, that is pitocin. Okay. So, here this is the process, and the whole and soul process again in the diagrammatic format it is given here. So, the diagram is saying us the process will start with a phase called dilation phase. Now, what you can observe in dilation phase, under influence of relaxin, the birth canal will get relaxed or dilated, that is called dilation phase. The mother body is preparing to expel out that baby out of the uterus. The second stage, that is the expulsion phase, under influence of oxytocin and prostaglandin, the baby will be thrown out of the mother body, that is called expulsion of the baby or the parturition process. And the third phase is the placental phase. Now, what happens here? Once the baby is expelled out from the uterine cavity, still the connection of the fetus with the placenta is there in the form of umbilical cord. Now, it is the time you need to expel out that umbilical cord as well as the placenta. So, after the delivery of the baby, it will take another 15 to 20 minutes of time, and the placenta will be kicked out from the uterine cavity, and this process we are calling it placental expulsion. Now, once the baby is delivered, now what will happen? The respiratory system, you need to start the respiratory system of the baby, and for that various hormones, the vasodilator like nitrous oxide will be produced inside the baby's body, and automatically there will be switch over, and the lungs and the respiratory system will start working, and then you can hear the first cry of the baby. So, after birth you can hear the babies crying. Why so? Because for the first time is inhaling or exhaling the air, and during the time of first exhalation, the baby will start crying by producing sound. Okay. Now, there is a important duty of the mother, that is to provide nourishment to that newly born infant, and the process we will be calling it lactation process. So, what is lactation? The process of formation and ejection of milk. So, the mammary glands of the female undergo differentiation during the time of pregnancy and start producing milk towards the end of the pregnancy. So, milk production started prior to the parturition by the process called lactation. Okay. This helps the mother in feeding the newborn. Now, which hormone is necessary for production of milk? Prolactin. That is also called as LTH, luteotropic hormone. Prolactin, that is responsible for formation of milk or production of milk into the mammary alveolar tissue. Okay. Another hormone is required, there is oxytocin, that will be helping the squeezing of the breast muscles surrounding the mammary alveoli for ejection of the milk. Okay. Now, once the milk is produced during the initial few days, initial 3 to 4 days, the milk is very much nutritious. That is called, that yellow colored milk is called colostrum, which contains several antibodies. Okay, that colostrum contains antibodies like IgA. IgA is present in colostrum, which will be providing the initial immunity, okay, initial immunity to the fetus, because the newborn baby, it doesn't have the essential immune system or the antibodies present inside the body. So that's why the mother will be helping that newborn to gain some immunity. Okay, so that is a kind of passive immunity, right? And also that colostrum is rich in proteins, and in compared to normal milk, it has less fats. Okay, now these antibodies, which are absolutely essential to the development, the resistance for the newborn babies. Okay, and which hormone will be helping into the milk ejection? Oxytocin. Help in ejection of milk. Okay, and which are the hormones which help in the development of mammary gland and the differentiation? Estrogen, progesterone, and human placental lactogen. Estrogen, what is the function of estrogen? That will be developing the mammary gland. Estrogen helps the female to develop the mammary gland. Okay, progesterone, that will help the mammary gland to develop the mammary alveolar structure in future, which are going to produce the milk while lactation going to take place. And human placental lactogen will help in the differentiation of a non-lactating mammary gland towards the lactating one. Okay, so this was all about this chapter. Now let's solve some questions, important question. So after formation of zygote, the question is given, after formation of zygote, it cleaves into 2, 4, 8, 16 daughter cells. Okay, so the daughter cell, this, after the cell division in zygote, what we are calling cleavage, we call them blastomeres. You know, we have studied the difference between the cleavage and mitosis. In mitosis, if this is the mother cell, two new daughter cells will be formed. If the mother cell is 2n, means diploid, the daughter cells are also diploid, but the cell size will be similar to the mother one. But in case of cleavage, if this is the zygote, we are considering this as mother cell, upon division, the cell will decrease the size, though the chromosomal set remains the same. So that is the difference between cleavage and mitosis. And for cleavage, it needs less time in compared to mitosis, because in cleavage, which stages are absent? In cleavage, there will not be any G1 and G2 phase of interphase. G1, growth 1 and growth 2 phases, they are absent in cleavage, and all the phases of interphase like G1, S and G2, they are present in normal mitosis. Okay. Now next question, assertion and reason. They are saying in humans, the male gamete determines the sex of the child. Yes, it's not the female gamete, because males are heterogametic, they can produce X sperm and Y sperm. So it's about choosing between X and Y. The determination of sex in human is polygenic trait influenced by genes of on the X chromosome. So though the assertion is true. The male gamete determines the sex of child, but the reason is false. So we can see that assertion is true but reason is false. So we will be ending our today's lecture with this question. So the question here is asking that which of the following hormones is responsible for fetal ejection reflex? Yes, and the answer you know, that is oxytocin. Okay, oxytocin hormone is related to fetal ejection reflex. When the signal is coming, neuroendocrine signal, then mother pituitary will start releasing oxytocin, and that oxytocin will act on uterine muscle for further contraction. Okay, so this is how we are at the concluding part of the chapter of like class 12, the first chapter of zoology, that is human reproduction. And I think each and every concept was catered properly. If there is any doubt, please comment in the below box. I will make a separate video on that. And obviously, all the related DPPs, practice sheets, and the handwritten notes of this particular class, we are gonna get at the PW app. And please click on the link given in the description box, that will redirect you towards the PW app. Okay, and please enroll there to get all the lectures, the notifications related to lectures. And for the time being, it's huge thank you to everyone, and bye bye and Jai Hind.